to start. All right.
All right. Excuse me. Yeah, welcome to the first MEMIS revision lecture. We will be starting off with biochemistry. Let's share screen. Okay, just a disclaimer. If you come to the Anki session, you probably know that I don't really take much notes. So these slides are remixed from last year's Enzyme revision lecture, last year's SWOT-VAC revision lecture, and PS Popper slides. Let's get started. Biochemistry. Okay. Sorry to be too in your face, but biochemistry is a pretty high yield topic. These are stats from, I think, 2019 or 2020's exams. As you can see, 24% of exam marks come from biochem. That being said, a lot of them are from metabolism, which will be up next. This is kind of an intro into metabolism to get you started. Yeah, an overview of lectures. An overview of this lecture. If you've been watching your lectures, you know osmolarity, tonicity, acid base, energetics, enzymes, cell signaling, and finally to intro into metabolism will be macromolecules. All right, let's get started. Fluid distribution. So 60% of your body is water, less in females. Out of that, 2/3 is intracellular, so in all the cells in your cytoplasm and all your organelles, and then 1/3 is extracellular, out of which there's interstitial, which is between cells. 80% of that extracellular, or 28% of total body water, is interstitial, and 20% is in plasma, which is in your blood stream. So just know it kind of just makes sense intuitively when you think of a space in your body. There's more cells than there is extracellular space, like because cells are very compact. So it kind of makes sense that there's more intracellular water than extracellular. So just think of that. And as we'll get into with tonicity, fluid does move between the extracellular fluid and intracellular fluid to maintain equilibrium or homeostasis. Next slide. All right. Osmolarity versus tonicity. This is a very high yield topic. You want to be able to know the difference. So let's work through it step by step. Osmoles is the total number of all solutes in a solvent. So it's just the number of any solute in a liquid, right? And it's millimoles. And then osmolarity is the total osmoles per volume, and that's usually in liters. So it's basically just millimoles over liters, and the solution is usually water again. And so where the distinction comes is osmolarity includes both diffusible and non-diffusible substances. So when we have a cell membrane, it's a semi-permeable membrane, and some molecules can diffuse, some can't. So osmolarity includes both of those. Sorry, oh, no. Sorry. Tonicity is something called effective osmoles, which is osmoles that cannot pass a permeable membrane, essentially impermeable molecules. And so when calculating tonicity, particles that can cross a membrane, which we'll go through in the next slide or in the next few slides, they're not included. Again, this is just a summary up here. Tonicity considers whether solutes are diffusible or non-diffusible, and osmolarity just includes all solutes. Now, let's go next slides. All right, again, to emphasize, because this is very high yield, they're distinct concepts. They're not the same, but they're related. So two solutions can be isoosmolar, but not isotonic. And now these are some terminology. So hypertonic, isotonic, and hypotonic. So this almost always refers to the extracellular solution. So whether the extracellular solution has higher, equal, or lower effective osmoles. And so that will lead us into osmosis soon. So basically this determines whether water will flow from inside to outside the cell or outside to inside the cell or maintain equilibrium. Diffusion is the passive movement of a substance from a region of high concentration to a region of low concentration. High concentration or low concentration, this will keep coming up throughout your entire med school career. And just know that particles move from high concentration to low concentration, barring one, which is osmosis, which we'll get into. It's a cool meme. Get it? It's a cell wall. And then this is passive transfer versus active transport, which main differentiation is ATP, which we'll get into soon. Now, here's what I was talking about with diffusible. Sorry, let me just pull the chat up in case. Okay, perfect. Membrane permeability. So we talked about when comparing osmolarity and tonicity, it depends on whether or not a substance can move across or can diffuse across a plasma membrane. And so hydrophobic molecules such as oxygen, carbon dioxide, nitrogen, etc., can easily pass through. Small uncharged polar molecules such as water, urea, glycerol can somewhat pass through. With your urea, just assume it can easily diffuse, but it's like a 50-50. And then basically as you go down this list, they become less and less permeable or less and less able to pass the membrane. So what we want to keep in mind is ions such as hydrogen ions, sodium ions, calcium ions, etc., cannot just diffuse past a plasma membrane. And so they contribute to tonicity, whereas hydrophobic molecules such as oxygen, carbon dioxide don't contribute to tonicity because they can easily diffuse through. They contribute to osmolarity. Now, this is the one exception. So osmosis, just get the definition down. It's very high yield. The net passive movement of water molecules from a region of low solute concentration to a region of high solute concentration through a semi-permeable membrane. They may try and trick you with the semi-permeable membrane. Just omit that. That's necessary for the definition, as you can see here. So this is the one time where you'll have something moving from low solute concentration to a region of high solute concentration. Just because that's the way it's defined, it's technically a region of high water concentration to a region of low water concentration, but we don't talk about concentrations of water. So it's low solute concentration to high solute concentration, as you can see in this diagram below. And this process is influenced by tonicity, which is a concentration, again, of non-diffusible solutes, because it's those non-diffusibles that create that imbalance and concentration gradient. I don't know if you've had Chris Wright's lecture. He'll talk about various theories as to why this happens on a very, very, very microscopic level, but I don't think you need to know that. So yeah, water will move in the compartment with the higher tonicity. All right, osmotic pressure, it's a bit of a weird topic to wrap your head around, but it's basically the pressure needed to stop osmosis. Right, so we often think of this U-shaped tube with a semi-permeable membrane in the middle. And basically the water is trying to get from the left side to the right side like this. And if you were to kind of just put your hand down on the right side and stop osmosis, that would be the osmotic pressure. Now, it depends on concentration and temperature, which is Van Hoff's law. I don't remember learning that. It was in the slides, but primarily it depends on concentration. So as you can imagine, if there's more solutes here on the right side, the water wants to diffuse across, wants to go through osmosis a lot more. And so the osmotic pressure would be greater. So the osmotic pressure, is proportional to the number of particles per unit volume of the solvent. And now the differentiation is it doesn't matter on the type, valence, weight of particles. It's literally just the number of particles in the solution. All right. Now here's when you get some clinical applications of tonicity. We talk about hypertonic, isotonic, hypotonic. This is just a visualization. So if there are more solutes in the extracellular fluid in this left picture, then water will move out of the cells because osmosis is water going towards the solution with the higher tonicity. If it's isotonic, water will move in and out. Now, bear in mind that water is still moving, water is still diffusing through the plasma membrane. It's just at equal rates in and out, which is equilibrium. And hypotonic, if there are less solutes in the extracellular fluid, water will enter the red blood cell, and then the red blood cell will grow and at one point burst, which is called cytolysis. So over here is cytolysis when the red blood cells burst in a hypotonic solution, and over here is crenation in a hypertonic solution when water leaves the red blood cells and shrivels up. Now, we need to know how to here before we get deeper into saline and dextrose and all, is red blood cells have a tonicity of 300 milliosmoles a liter. Just remember 300, because that'll be important coming up. Now, this is a sentence faculty says a lot, and it can be a weird existential thing to think about, but your red blood cells sit in an infinite tub of fluid. Now, what this means is the extracellular volume, if you think of all the extracellular volume in your body, which includes your interstitial fluid and your plasma, is so much larger than a single red blood cell's volume. If you think of a single red blood cell, it's tiny, it's microscopic. And all the fluid in your body is so much larger than that they just call it an infinite tub of fluid. And what this means is that a slight difference in tonicity in your plasma fluid will cause a huge change in the red blood cell, because it's based on concentration, right? So if the concentrations were to equalize, it's obviously a lot more solutes in the huge extracellular fluid than it is in the single red blood cell. So you'll get questions where you're injecting something into a person in an IV, and you want to see what impacts that has on their red blood cells in their bloodstream. And so this is a table just to set it up, but we'll go through it. And there's a huge summary table at the end. But basically, if you think about injecting pure water, right? We know that the red blood cells are 300 milliosmoles a liter in terms of tonicity. And pure water has no solutes in it whatsoever. So then pure water is a hypotonic solution, and water will move into the cells and cause them to increase in volume and explode. So if you think about it, we write down initial osmolarities, which in the intracellular fluid of the red blood cells 300. In the solution is 0 because it's pure water. And we know that none of these have diffusibles that we're taking into account. And so the final osmolarities are the same. And the difference in this row of the table, 300 versus 0, is what determines whether or not water moves into, out of, or stays constant between the cell and the ECF. The pure water is pretty straightforward. If you think of urea, now urea, if we go back, can diffuse through the plasma membrane. So it doesn't contribute to tonicity, it contributes to osmolarity. Now, initial osmolarities, both again are 300, and diffusibles move to equilibrium. And, this is saline. Here, urea. So urea can diffuse into cell. And because we think of it as an infinite tub, we assume just when we think of the final osmolarity, the concentration of the intracellular fluid just doubles while the solution is unchanged because there's just so much of it. So you think of the final osmolarity in the red blood cell, and it's 300 plus 300, so 600 milliosmoles a liter, while the extracellular fluid of urea is just 300. And as you can imagine, this would be catastrophic because all the water from the solution again would move into the red blood cell. Now, this is something that never, ever happens clinically. Faculty just likes to test you on it, to test your understanding of tonicity and osmolarity, but never, ever inject urea into a patient. Normal saline. Now, saline, 0.9%. Keep this in mind. They'll try to trick you with 9% or 0.09%. 0.9% saline being injected into the extracellular fluid will be ideal because it also has a tonicity of 300 milliosmoles per liter. So as you can see here with final osmolarity, so water doesn't move in or out. It is isoosmolar and isotonic, whereas urea is isoosmolar, but it's hypotonic because it can diffuse. These are just some calculations. You probably may have exam questions where they don't present it to you in percentages. They'll present it to you in like 1.8 grams per 100 ml of urea and stuff. So you'll have to ideally convert it. And so this is just calculations on how to convert it. And so this is a big summary table. Dextrose. There's no slide on dextrose. Okay, I'll go through dextrose as well. But pure water, saline, and urea we've been through. This is, just get this table down. It makes sense once you start thinking about it. Now dextrose is a bit different. It's something we use clinically. Dextrose is basically just glucose, and it's 5% dextrose. So initially, it is isotonic compared to the 300 milliosmoles in the red blood cell. And so there is no net water movement. But when, because it's glucose, as you'll learn in the next lecture, the dextrose gets metabolized by the cell. And so then what happens is the extracellular tonicity decreases, and then water gradually moves into the cell. Yeah. So as the dextrose in the extracellular fluid is being metabolized, it goes from 5% down 4, 3, 2%. But this happens over a long period of time. So there's no cytolysis. And so how this table, what's new that is worth knowing is that when you inject saline, your extracellular fluid increases. And when you inject dextrose, your extracellular and intracellular fluid increases. So your total body water increases. That's something they can test you on. All right, acid-base physiology. Yep, memorize these numbers. So neutral pH, I know we learned neutral pH is 7. In the body, it's 6.8 because of the temperature at 25 degrees Celsius. So neutral body pH is 6.8. But normal body pH, which is what we want, is 7.4. The range that's acceptable is 7.36 to 7.44. So what that means is we want our body to be a bit alkalotic. Basic, yeah. Now, acidosis and alkalosis, which are terms that come up a lot clinically, are in reference to normal body pH. So 7.4. So if it's below 7.4, it's acidosis, and if it's above 7.4, it's alkalosis. Excess CO2 makes the blood acidic. You've probably done this in chemistry, where it turns into bicarbonate ions and hydrogen ions, and that increases the acidity. And buffering mechanisms is, yeah, bicarbonate carbon dioxide mechanism. You guys will get through this next year. It'll be fun. All right, moving on to energy and thermodynamics. Two types of energy, potential and kinetic energy. Potential energy is energy stored in the chemical bonds, in concentration, in gradients. The way we learn it is like if you hold a ball up above the ground, it has potential energy because it can fall. And then kinetic energy is the energy of motion. So when you drop the ball, it's moving, and that's kinetic energy. Two laws of thermodynamics that we need to know is first law, energy can't be created or destroyed. It's always converted between different forms. And second law, based on that conversion, is it's not 100% efficient. And some of that energy is lost, usually to heat, but sometimes to other forms of energy. Calculating energy. So I don't believe we've ever been asked to calculate it, but I guess it's just useful to know if you want some extra knowledge. G equals H minus TS. I'm pretty sure it's usually delta G equals delta H minus T delta S. Yeah, we don't need to. But what that's important for is when you calculate this, delta G determines whether a reaction is exergonic or endergonic, or you may have learned exothermic and endothermic, basically the same thing. Exergonic reactions are spontaneous. They want to happen, and they release free energy. They have a negative delta G value, and they break things down, which is what catabolic means. When you break a bond, basically, that bond has energy in it. That chemical bond has energy in it, and when you break it, you release that. Anabolic reactions are when you're building things up, are endergonic. They have a positive delta G, and they're non-spontaneous. They use energy. If you think of bonds as having energy, then if you break a bond, it's going to release energy, and if you make a bond, you're going to need to put energy into that. And so another way to figure out delta G of reaction is delta G of products. Mg of reactants. This is just a table. So we were thinking products minus reactants, right? So this is a table where we have Gibbs free energy, which is G on the y-axis, and time on the right axis. So as a reaction is happening, if this is the activation energy, this little hump, which is the minimum amount of energy you need for a reaction to take place. When you think of products minus reactants, if the products end up with more energy than the reactants, that's endergonic. So think you're building stuff, you're making bonds, you're putting energy in, and reaction is not spontaneous. If you think of exergonic reactions, the products have less energy than the reactants because they're releasing energy and the reaction is spontaneous. And these diagrams will come back up when we do enzymes in a bit. All right, ATP, adenosine triphosphate. So it's a nucleotide. If you've done your central dogma and stuff, you know adenine and ribose and a chain of 3 phosphate groups. So here's a little diagram of adenosine, ribose, and 3 phosphates. And basically between phosphate 2 and phosphate 3 is a very high energy bond. So they refer to ATP as the currency of the cell. It is what fuels a lot of the reactions in our body. And so when we need energy in our body, we break this high energy bond using an enzyme, and that releases energy, catabolic reaction, exergonic reaction. And that essentially leaves us with this 1 phosphate group and ADP, adenosine diphosphate. Diphosphate with 2, triphosphate with 3. It can also phosphorylate molecules. We don't go through that too much. It's not very high yield. But yeah, adenosine, ATP, very high yield. Here's another representation. You break this bond between the 2nd and the 3rd phosphate group, and you're left with energy, inorganic phosphate, and ADP. All right, moving on to enzymes. We talked about this little diagram with the progress of reactions. We have all these reactions happening in our body, but we need to speed them up. And so anything that speeds up a reaction is a catalyst. Biological catalysts made up of proteins are enzymes. And if you think of the one major thing they do is they lower the activation energy by providing an alternate reaction pathway. And what this does is if there's less energy required for a reaction to take place, it increases the rate of reaction because it happens more often. So here you can see that your activation energy, we talked about this initial hump, is lowered by an enzyme. And what's important to note is that they don't end up changing your delta G. So your reactants start in the same place and they finish in the same place with regards to free energy. So the difference is the same. They have specific conditions for function. All throughout our body, we have places with different pHs. If you think of our stomach is very acidic, the bloodstream is relatively basic. So enzymes that exist in those areas can only really exist in those areas. And temperature, we have body temperature. So as you can imagine, all the enzymes in our body work ideally at our body temperature. And I forgot to put the graphs, which basically show the enzyme, how enzymes function at different temperatures and different pHs. I'll put those in the slides after. If not, we went through it in PS Pinata. You can check that out, I believe week 2. All right, enzyme inhibition. When we need to control how enzymes work, this will also get you a bit into pharmacology. This is also how some drugs work. So you have these 3 major types of inhibition, which are shown by these diagrams. This pretty much sums it up when you're not thinking, when you have to think about the high yield stuff, but let's go through it a bit deeper. So competitive inhibition. We know that active sites on enzymes have a very specific formation that's very specific to the substrate, right? And so if a competitive inhibitor comes along with a very similar shape to the substrate, it can actually bind to the active site. And if the competitive inhibitor is binding to the active site, the substrate can't. And so that blocks off the enzyme's ability to react on the substrate. However, if you want to overcome the effects of that inhibitor, you can increase the substrate concentration. And eventually, because they're competing, if there's more substrate, the competitive inhibitor stops working. Non-competitive inhibitor binds to an alternate site, which is called the allosteric site on the enzyme. And when it binds to the other side, it changes the conformation of the active site. And so the substrate, again, can no longer bind. And how this is different primarily is that you cannot overcome this by increasing substrate concentration. So in this graph, on the x-axis, we are increasing substrate concentration, and over time, the competitive inhibitor will come to the same rate of reaction as the normal enzyme, whereas the non-competitive inhibitor will stay at a decreased rate of reaction. Uncompetitive inhibitor will bind when the enzyme and the substrate form a complex. So after the substrate has attached to the active site, that's when the uncompetitive inhibitor comes on and prevents the substrate from being released, the product from being released, okay? So again, if you think about this diagram, competitive inhibitors are going right to the active site, non-competitive are going to an allosteric site, and uncompetitive attach after the substrate is attached and prevent it from being released. Lastly, negative feedback pathway. So this is something you'll see a lot in your body. I think even with alcohol metabolism, there's an aspect of this. And so it involves allosteric inhibition. So when you think of these different pathways in our body, there's almost never just one enzyme. It's usually a whole chain reaction of enzymes where the product of enzyme A will be the reactant of enzyme B. So here we have A, B, C. And so if this is the end product, let's say this whole chain is producing D, when there's enough of this end product D, it'll actually go back up to the enzyme, to the first enzyme over here, bind to an allosteric site, and inhibit that, change the active site and inhibit the substrate from binding. So it's a really neat way to regulate homeostasis in our body. If there's too much of this end product, it can actually go back to the beginning of the pathway, or the middle of the pathway, somewhere in the pathway, and block that off so that there's no more end product being created. If you guys have any questions, just chuck them in the chat. I've got it open. Cell signaling. Relatively low yields, but there's these different types. So contact-dependent cell signaling is when your two cells that need to talk to each other are directly touching. An example, T cells and target cell. We get into that with immunology. Synaptic signaling is between neurons. So neurotransmitters have a synaptic cleft or a small gap, which is also similar to paracrine signaling, where signal is transmitted to the nearby environment, okay? So if two cells need to talk, they can literally directly touch and talk, which is contact dependent. They can release a signal into the surrounding environment just nearby, that's paracrine. Endocrine is the signal gets sent into the bloodstream and can affect cells all throughout the body, far away. And autocrine is a cell will secrete a signal that'll affect the same cell. So it's a bit difficult to think about, but I think visually it makes a lot of sense. Here's contact dependent. The signaling cell and the target cell are touching with this membrane receptor. Paracrine is nearby cells are being signaled. Endocrine is it enters the bloodstream and can travel all across the body. And synaptic is to do with neurotransmitters. All right. Now, as your final segue from biochem into metabolism, we'll go through some macromolecules. So carbohydrates, carbo and hydrate is consisting of C, H, and O. Hydrate is H2O. Carbon is carbon. Monosaccharide, disaccharide, all this is basically just increasing units. So if you think of glucose and fructose and galactose as your units, as you build them up, they go from mono to di to oligo to polysaccharides. Honestly, all you really need to know is glucose as a monosaccharide, and then polysaccharides, we'll get into them soon. Those can be pretty high yield. here are your functions. Energy storage. Number one, glucose provides us energy, and that will lead into metabolism with glycolysis and all that. Providing bulk is fiber, cellulose, which is a polysaccharide. And all this, yeah, you can go through these functions. All right, polysaccharides, as promised. Starch is a polysaccharide, which has two different versions that we think about. We have amylopectin, which you can see over here, and we have amylose, which you can see over here. All starch is produced in plants and is digestible. What you need to know differentiate between these is amylopectin is occasionally branched and amylose is linear. And then on the other hand, glycogen is how we store glucose. So if you think of all these little beads as little glucose, you can see how huge these polysaccharides really are. And so glycogen, highly branched, amylopectin, moderately branched, and amylose, linear. We've had exam questions on these before. Now, if you want to think of branching, these are your glucose molecules. You have an alpha 1,4 glycosidic bond. As you can see, that's linear because just the bonds that are being created between which carbons, it's a linear bond, and that's what you'll see in amylose and some of amylopectin. And then basically what contributes to bonding are these alpha 1,6 glycosidic bonds. So you have this chain, and then you're adding another chain, and this vertical bond is a 1,6. The 1,4, 1,6, that just refers to which carbons are being bonded. And lastly, cellulose is produced in plants and it's indigestible. That's what we know as fiber. Kind of just passes through our system. We can't absorb it. All right, lipids. Sorry, we'll do this first. You have three major types of lipids. You have your tags, your triacylglycerols, so which is your 3 fatty acid chains. Do we have pictures? Yeah. Okay. 3 fatty acid chains over here, 1, 2, 3, with a glycerol. And that's your primary form of energy storage because you get into this in metabolism, but fats are very, very efficient form of storage. They can hold a lot of energy in all their bonds. And so this is our major dietary source of lipids, are these tags, right? Then we have phospholipids, which are in our plasma membranes. If you remember, we have the bilayer of phospholipids. It's basically a phosphate head over here. I think phosphate, we also saw that in ATP, so a lot of renewable molecules. A glycerol, and then 2 fatty acid chains. Now, what this is unique for is that it has this hydrophilic head, which is with the phosphate, and this hydrophobic tail, which again, you guys know about this in plasma membranes. And lastly, steroids. A very unique structure because they have these 4 fused hydrocarbon rings, right? Precursors for hormones, bile acids, vitamin D, very important in our bodies, and also a component of cell membrane fluidity, right? We have our cell membrane and it can move, it has that fluid mosaic model. So steroids are what help do that. And on that note, one type of steroid, think steroids as ending in sterol. So cholesterol is that one type of steroid that's important for your cell membrane. And it also is a precursor for testosterone and cortisol, as you can imagine, very important hormones in our body, bile acids, and vitamin D. All right, that's lipids. Proteins are extremely versatile molecules, macromolecules. These are all their functions, and they'll keep coming up again and again, so don't stress. You'll talk about DNA and genetics. You'll talk about antibodies and immune function, digestive enzymes. That's what we're doing now in year 2. Muscle movement and contraction, support for the body, hormones, and they help move essential molecules around the body. So think hemoglobin as a protein that moves oxygen around our body. All right, basically 4 different structures. It's not the highest yield, but you have primary structure, which is just a linear sequence of amino acids, which is formed through peptide bonds. Think condensation reaction. If you're making bonds, you need to put energy in it, non-spontaneous anabolic reactions with a delta G being positive. That's how these primary structures are made, which are just linear. Over here, you can see these linear chains of amino acids. Secondary structures, you start folding this line of amino acids, interactions with the polypeptide backbone. So this is the polypeptide backbone. And as these interact, you get these alpha helices, which you can see over here, these little spiral things, and beta pleated sheets, which is kind of a twisting motion, but it's like flat sheets. These are the two structures in secondary. Here's another picture of beta pleated and alpha helix. Tertiary structures are when you get more bonding and more interactions, and that forms a 3D structure. And folding is due to interaction between the R groups. So with the protein with the protein molecule, amino acids, there's an R group, and when these interact with ionic hydrogen bonding disulfide bridges, they form these folds that make a three-dimensional structure. And lastly, a quaternary structure, or fourth degree, is multiple polypeptide chains, usually held through with hydrogen bonding. They're quite similar to tertiary structures, just there's multiple of them. Classic example of this is hemoglobin. It has 4 polypeptide chains, and you see it in your blood for transport of oxygen, etc. And that is all we have for today. So let me just stop sharing. For questions, for your questions, put them in the chat. For questions to quiz yourselves on, you've got the academic drive. There's a heap of questions on P.S. Popper's end of sem extravaganza and last year's revision lectures. But yeah, you can go through those. Best way to learn. And any questions, just chuck them in the chat. If not, over to you, Yash, if you're ready. You're in mute.
Oh, sorry. All right. Well, thank you so much, Ada, for that amazing, amazing little revision lecture there. So I think Yash is here. Yash, are you ready to start? Or otherwise, actually, we can give everyone like a 10-minute break or something.
Yeah, I reckon that could be good, and then we can get started.
All right, perfect. So if you guys want to take a quick 10-minute break, we'll start at 10. We'll start at 11 o'clock basically, 10:55, around about that.
Yeah, sounds good.
If you guys have any questions about things in general, content even, feel free to shoot them through. But yeah, we'll see you guys in 10:15. All right, everyone. And we're back. So I'm gonna hand it over to Yash to take you guys for the metabolism revision lecture.
Perfect. Thanks, Vivi. So before we get started, hold on, let me just quickly split screen. Yeah, no, I'd just like to sort of begin by saying I encourage as many of you to sort of ask as many questions as you want, to possibly turn on your cameras or to unmute yourself at any time, just to make this as interactive as possible. I should probably share screen as well. Okay. Just making sure, Vivi, you can you see this?
Yep, all good.
Okay, perfect. Alrighty. Yeah, so sort of like what I was saying, I encourage you to sort of be as interactive as possible. Personally, I found metabolism to be one of those topics last year that was a little confusing. I remember with all the sort of NERMAs, active learning sessions, I was just really confused. However, I found the revision lectures to be particularly useful. So again, my sort of motive behind doing all this is to make it as clear and as simple as possible. So a few credits before I begin, which is super important. One is PS Python. The next one is faculty and all the resources they give us. The other one is Raman Sanjayan from my year, who wrote some incredible notes on this topic. And I highly recommend all of you, if possible, to have a look at those as well. And also the 2019 Metabolism Revision Lecture. And another quick apology as well. I am a little under the weather, so my voice may seem a little nasally. So if you do have trouble understanding anything, please put into the chat, and I'm happy to repeat it. So yeah. All right, so again, before we begin, I'd like to provide a bit of a short spiel on a bit about what I'm going to go through today. So I want to keep the content and I'll try to keep the content as exam focused as possible. Metabolism, like I said, is one of those topics which you can go into depth for 1,000 years. So I want to make it as exam focused as possible. And for that reason, I'm going to be including I've included at the end of the presentation, close to 20 ish past exam style questions. So please feel free to stick around for that. And again, I'd just like to say metabolism is quite a high yield topic. So yeah, it's really important that you have a good understanding and a good grasp over it, so that you perform well in your end of semester exams. Okay, so a bit of an overview of what I'll be covering today. Firstly, it will be carbohydrates, then it will be respiration, both anaerobic and aerobic. Next, it will be lipids, then alcohol metabolism, a bit on amino acids, then energy sources of cells, and the great metabolic race. And again, all throughout, I'll be pointing out what sort of information is important and what information you can have a read of in your own time. Okay, so I'll quickly just do one thing as well. I think this presentation is a little outdated, so I'll just quickly hit refresh and then I'll share screen again. So bear with me for one moment. Okay, perfect. Present to view. Sorry, guys, just one moment. Okay, all right, should be good to go. Pause, reset. Okay, all right, I'll have a go at sharing screen again. Okay, lovely. So let's get into it. So done that. Okay, so a bit on carbohydrates. So I know Yachat has gone through a bit on sort of macromolecules, but again, I'll just provide a bit of a 3-4 chem refresher. So on the left, you have glucose. It's a monomer. It's a 6-carbon sugar. It's the form that is absorbed through the small intestine, which I'll be going through in a bit more detail, and can be transported in your blood. On the right, you have glycogen. It's highly branched. It's a polysaccharide. It's a polymer of glucose. It's used for storage. and a bit on its structure as well. So its high branching allows for a larger surface area and therefore results in more rapid breakdown, which we'll go into a bit more as well. And like Yajat mentioned very briefly as well, it contains two types of bonding. One is the alpha-1,4 glycosidic bonds and the alpha-1,6 glycosidic bonds. And next, okay, so a few key definitions as well, just because I guess with metabolism, there are a lot of terms that may sound similar. So again, just trying to break it down into as simple as possible so you understand it. So a key thing that I used last year was anything that says glycogen concerns glycogen. So if you can see here, you have glycogenolysis, that concerns the breakdown of glycogen. Here you have glycogenesis, which is the creation of glycogen. And here you have glycolysis, which is the breakdown of glucose into two pyruvate molecules. And then you have gluconeogenesis, which is the creation of glucose from non-carbohydrates. And I'm sure all of you know this already, but if you have lysis, it sort of concerns the breakdown of molecules. And then here, if you have genesis, it sort of talks about the creation. Okay, so the way I really wanted to break this down was into three stages, right? You have your digestion, you have your storage, and then you have your mobilization. Now, if you look at the digestion of carbohydrates, let's use the example of a donut, right? So you eat your donut and it starts in your mouth where your salivary amylase breaks down those alpha-1,4 bonds. And then, your donut moves down into your abdomen and then it continues into the small intestine where your amylase, again, sort of breaks it down. So your amylase breaks down the starch to simple sugars. So it goes from a polymer to a monomer, whereby then the monosaccharides are absorbed through the small intestine into the bloodstream. Okay, so that concerns digestion. Then we'll move on into storage. So when blood glucose levels are high, after we've eaten that donut, we have something that's produced and it's called insulin. And that signals for muscle and liver cells to uptake this blood glucose and store it as glycogen. And that's what we call glycogenesis. Whereas when blood glucose levels are low, your body releases something called glucagon, and that signals muscles and liver cells to break down that stored glycogen into glucose for our body to use. And that's called glycogenolysis. And then lastly, we have mobilization. So when needed, this glycogen can be mobilized and it is converted to glucose 1-phosphate. So your muscle cells, they really want this energy pretty quickly. So the muscle cells, they further convert this into glucose 6-phosphate and they respire this straight away. Whereas in the in your liver wants to export this energy. So in the liver, glucose 6-phosphate, is converted into glucose by the enzyme glucose 6-phosphatase. And then this free glucose enters the bloodstream and can be transported to other cells. Okay, moving on. So now we'll be talking about gluconeogenesis, right? And this process occurs, this process of creating glucose from carbohydrates occurs only in the liver, and it is used during fasting/starvation when all carbohydrate stores have been depleted. So here you can find a few sort of sources that are altered to form a reactant in one of the steps of glycolysis and the citric acid cycle. So you have glycerol, which feeds into step 5 of glycolysis. You have lactate that feeds into pyruvate. You have acetone that feeds into pyruvate. You have amino acids, which feed into pyruvate and some of the citric acid cycle once deaminated. And you have acetyl CoA, which is already in the cycle. So if you were to really simplify it, it's basically glycolysis in reverse to make glucose rather than break it down. And the reason we have this process of creating glucose is there are certain cells in the body that only use glucose. I'll be going through this later in the presentation, but for an example, that would be your red blood cells, which is why sort of when your dietary glucose levels are depleted, it's important for the body to be able to produce its own glucose to continue sort of the metabolic processes, which is why we have gluconeogenesis. So glycolysis, another thing to sort of note is glycolysis is totally reversible, except you cannot get pyruvate from acetyl-CoA. So acetyl-CoA has to go all the way around the citric acid cycle to oxaloacetate, and then skip pyruvate on the way back up to go to glucose. And this is shown pretty clearly in the next two photos that we have here. So as you can see, the first steps here, if you can see my mouse, are completely reversible. However, when you hit pyruvate, it isn't reversible. So when you go to acetyl-CoA, you have to go through this entire cycle and go to an intermediate, and then you can go all the way back up to glucose. So this is pretty much your body bypassing this little downward arrow situation, if you were to put it simply. So here, the image on the top left shows a few more things that we've talked about sort of feed into the cycle. And the image on the right is your source of blood glucose throughout the day. And I think it's really important that we go through this in a bit of depth. So firstly, let's go through your dietary carb intake, right? So here you have your breakfast. Yep, so you eat your donut, and here you can see that your blood glucose levels have a bit of a spike and they go up. And then as you use it, again, they sort of decrease. And then again at lunch, they increase and then they decrease. And then again at dinner, they increase and then they decrease. I think that's pretty straightforward. Then we'll move on to glycogenesis, right? So here, this is the purple. So as you can see with, with glycogenesis, yeah. So as you can see with your glycogenesis, so this is when your sort of glycogen levels spike. They spike when your dietary blood glucose levels are sort of on the down, right? So it still needs, you still need that glucose. And so what your body does is it starts to break down your stored glycogen, which is why, again, you see this spike in glycogen levels before they, again, drop. And then the question you'd probably ask is, why do you sort of have this sort of plateau of glycogen levels before breakfast? And the reason for that is that over your sort of sleep, your body is in a starved state. So you haven't had that, you've had that last donut at 6 p.m. the night before. So what your body's done is that it's sort of utilizing your stored glycogen. And then over the morning, as you sort of begin to wake up, again, that the glycogen levels start to decrease. And then lastly, we have gluconeogenesis. So again, like I said, gluconeogenesis, it's only really important during starvation, which is why, again, before breakfast, you can see your body really sort of favors gluconeogenesis. You can see the spike. But again, after breakfast, it sort of becomes a bit smaller in terms of its usage. And again, in terms of gluconeogenesis, like, let's just say, so sort of from a first principle point of view, like, why wouldn't your body want gluconeogenesis when you're sort of having these meals? So let's just say one sort of way of producing glucose is to use your amino acids or your proteins, right? Or your muscle in your body. So if you are having these meals, your body, it's probably not in your body's best interest to be breaking down your muscle, right? Just from sort of a survival standpoint, which is why, again, your body's pretty smart and it sort of tries to limit gluconeogenesis whilst you're having meals, because you really don't need that, the breakdown of your muscles, and you really don't need that excess glucose as well, because you've got your glycogen and you're also having your dietary carbohydrate intake as well. Okay, so I hope I've made that pretty clear on your carbohydrates, and we'll be moving on to respiration. Okay, so I touched on a bit on glycolysis. So you have your glucose, and that's converted to 2 pyruvate molecules, but then you've got sort of 2 pathways here. One, you have your anaerobic respiration, which is where your pyruvate is then converted to lactate. It's incomplete oxidation. And then you have your aerobic respiration, where your pyruvate is converted to acetyl-CoA. And then your acetyl-CoA goes into the citric acid cycle. And then the products of that, which is your NADH and then your FADH2, they go into your electron transport chain and that's what we call complete oxidation. But we'll be going through this in a bit more depth, and this is just an overview. Okay, so I thought it was also important to talk a bit about the locations of metabolic processes, just because sometimes they like to test you in exam questions as well, where do certain sort of processes take place. So first we have glycolysis, where your glucose is converted into your 2-pyruvate molecules, and this occurs in the cytoplasm. And then you have your fermentation or your anaerobic respiration, and that also occurs in the cytoplasm. And then you have your pyruvate decarboxylation, which is pretty much your conversion of your pyruvate to your acetyl-CoA. That occurs in your mitochondrial matrix. Your citric acid cycle also occurs in your mitochondrial matrix. And then you have your electron transport chain, which is in your mitochondrial inner membrane. Again, what would I take away from this in terms of high yield? They really like to sort of ask, where does glycolysis take place? And also, where does the electron transport chain take place? So have a read of that before you sort of sit your exams. Okay, so again, we've gone through glycolysis, and you've probably gone through this in a lot more depth in your own learning and in your classes. But I'll tell you that this is a 10-step process, and it's really important that you note that you don't need to memorize it all. So even for me, what did I take note of, and what did I really remember last year? One is that the net total of this process is 2 ATP being produced per glucose molecule. And then overall, if you look at the products, your glucose gives you 2 pyruvate molecules, 2 ATP, and 2 NADH. And again, that's sort of all I'd really know in this process. I guess something really small is that your first step is you convert your glucose to your glucose 6-phosphate, which comes up in a few more slides as well. Okay, just checking the chat. Let me have a quick check of when these was asked. Okay, so the first one is, why does the body favor glycogenolysis over gluconeogenesis between meals? So Joshua, I hope I've answered this. So your gluconeogenesis is really only used when you're sort of starving and in your starved state, whereas and it sort of is your third sort of glucose producing step that your body likes. One is sort of your dietary carb intake, then you have your use of glycogen, and then you have your gluconeogenesis. And again, like I kind of wanted to point out, sort of from a survival standpoint, one of the processes of gluconeogenesis can be to use your amino acids or your proteins or your muscles, really. So again, from a survival standpoint, it probably isn't in your body's best interest for you to be breaking down your muscle, right? Which is why it's a bit more safer, a bit smarter for the body to utilize that stored glycogen that you have, and sort of converting that into your glucose. So Joshua, if you want to put into the chat or even unmute, does that kind of explain? Okay, perfect. No worries. So again, I really like to break it down into sort of really simple terms just to make you guys really understand it. And then I have a question in my private message as well. So I'll have a quick read of that. Okay, so I think the question is, yeah, okay, so, okay, yeah, so what you have to understand, and I think Yajat might, there's sort of, you have your non-reversible and then you have your reversible sort of reactions. And when you go from sort of your 5th step onwards, you'll find, and when you hit pyruvate, you'll find that the reactions aren't reversible. So you can't really go up back towards glucose. What you have to do then is you have to convert your pyruvate to your acetyl-CoA, go through this entire process, go to an intermediate, which again, the reason it's written intermediate is you don't really need to know it at your stage. So I wouldn't even recommend reading into it. And then you go from your intermediate back to this step where you can, again, then the reactions are reversible, and then you can go straight up to your glucose. So to the person who asked me that in the private message, does that kind of explain your question? If you want to just message me again, that would be good, and then I can move on. Yeah, perfect. Okay. Okay, so where were we? Yeah, okay. So now we'll be going through a bit of anaerobic sort of respiration. So what's important here is, okay, there's a lot on the slide. And again, I'd like to point out that I think there's a lot of information on these slides. I won't be going through a lot of it. I'll just be pointing out what you really need to know, because a lot of it will come up in sort of our alcohol metabolism when we talk about some of the pathologies that we see. Okay, so here we have our pyruvate, and that is converted to your lactate in the cell's cytoplasm, as I've said. What is important to note here, so when you convert your pyruvate to your lactate, this doesn't actually produce ATP. However, it does produce NAD+, which feeds into your glycolysis, which then produces true net ATP. So again, for that reason, it's not very energy efficient. Okay, and another thing to note is that when you go from your pyruvate to your lactate, the lactate is able to dissociate into lactic acid, which can lower your blood pH, which can cause lactic acidosis, which isn't great. And that will come up when we go through our alcohol metabolism as well. What a couple other things, again, I'd like to stress is that this process has an input of NADH, and then it sort of, one of its products is your NAD plus. So again, that's important. Okay, so then we'll be going into a bit of the Cori cycle, a bit of, and that's pretty much anaerobic respiration and a bit of gluconeogenesis. Okay, so like I've said here, we have our pyruvate molecule here, right? And there we convert it to our lactate, and that's our anaerobic respiration. So that's here on the right. So then we have this blood lactate here. And like I said, this lactate we can actually use to produce some glucose. So that's your gluconeogenesis. So here we convert our lactate to our pyruvate. And then to go from our pyruvate to our glucose, we actually have to have an input of ATP, and then we have our glucose. Okay, so let me quickly check my speaker notes. Is there anything else that I wanted to point out? No, not too much. So I guess another thing is that in muscles, when you have your anaerobic respiration, that's when your pyruvate is when you have your anaerobic respiration in your muscles. So this is on the right. That sort of has that creates your ATP. It gives off an ATP, but it also creates your lactate, and then that can, using gluconeogenesis, you can produce a bit of glucose as well. But again, Cori cycle, not too high yield. So yeah, I wouldn't worry too much. Okay, but then you have your aerobic respiration. So again, it's pretty energy efficient. So when O2 is available, when your oxygen is available, aerobic respiration is preferred. This takes place in the mitochondria, where pyruvate is converted to acetyl-CoA. What's important here is that it requires NAD+, and it produces some NADH. That's about all I think that is important. I think a lot of us know that our aerobic respiration as well. But again, point out, requires NAD+, produces NADH, and it's pretty much a conversion of your pyruvate to your acetyl-CoA. And again, like I've said, this process is actually your pyruvate decarboxylation as well. So if you see that buzzword, that's just your aerobic respiration of your pyruvate. So that's in, again, like I said, that is in your mitochondria. It takes place in the mitochondria. Okay, so again, citric acid cycle. You've probably seen a billion med memes of the citric acid cycle and the inherent need for us to memorize it. But again, like I just want to tell you, don't freak out, don't memorize it. Just know your sort of key takeaways, which is what I'll go through. So pretty simple. What feeds into the citric acid cycle? That's your acetyl CoA, yeah? And that interacts with your oxaloacetate to form your citric acid, hence the name. And again, all you really need to understand is the bottom line and what is really created. So through these steps, energy is produced, right? And it's in the form of this thing called GTP, and it's the same concept as ATP. But what else is produced? You have your NADH here, and you also have your FADH2, which are important as we move into the electron transport chain, right? So again, the slide here is way more detailed than you'll need to be concerned with. So again, I'll really point out what is important. So again, a real quick summary of what this entire sort of cycle is. So do you remember those substrates that I talked about that come off our citric acid cycle? So namely your sort of NADH and your FADH2. So these are actually, they're actually reduced to their lower energy states. And this energy of this transition to the lower energy states is used to then push hydrogen ions against their concentration gradient to the intermembrane space, right? And again, this is just your mitochondrial, So it goes into this intermembrane space, and this is your hydrogen ions. And then what happens after that? So your hydrogen ions here, yeah? And then they move back into the mitochondrial matrix through the ATP synthase pump, which is over here, where my mouse is. And the energy of the hydrogen ions, your H+ ions, moving down the concentration gradients through this ATP synthase pump is what is used to convert your ADP to your ATP. And voila, you've got your energy, finally. So again, real simple, your NADH and your FADH2 are reduced to the lower energy states. The energy of that transition is used to push these hydrogen ions into the intermembrane space, and then they move down the concentration gradient through this ATP synthase pump to convert your ADP to your ATP. Okay, all right, so just check the chat, there's no questions, which is a good sign. Okay, now this is a bit of a summary of what are the sort of products of your aerobic respiration and how much ATP is really created, right? So what's important here is you to know that your total ATP produced is 32. And what's the breakdown? So you have your 28 from your electron transport chain, you have 2 from your citric acid cycle, and you have 2 per glucose molecule from your glycolysis. This is another sort of concept that comes up in exams, which I want to stress. So when you go in your electron transport chain, when you have your 1 NADH to your NAD+, that gives us 2.5 ATP. So just know that for NADH, it's your 2.5, that's your sort of ratio. And when you have your FADH2, it's the ratio is 1.5. That's come up many a time in your past exams. So take a note of that as well. Okay, now we'll be transitioning to lipids, which in my opinion is somewhat easier. So let's have a go at it. Okay, so I know Yatjat kind of went through this as well in terms of your macromolecules. But yeah, I think again, in first year, having not done biology myself in year 12, I was easily confused by a lot of the medical sort of the jargon or the terms that were thrown around with lipids. So again, just to clear it up, and I'm sure all of you know it, but again, just for sort of simplicity's sake, your triacyl glycerols are the same as your tags, which is what I'll kind of be referring to them as I go through this lipid section. And they're the same as your triglycerides. So just take a note of that. And what are your sort of key lipids to take note of? Here you have your glycerol, you have your free fatty acids. So your glycerol with 3 fatty acid chains gives you your triglyceride, which is the same as your tag, which is the same as your glycerol. Then you have your ketone here and cholesterol, which are important as well, and I'll be going through in this next section. Okay, so again, like I did with carbohydrates, I think it's really important that we go through this in steps that make a bit of sense. So you have your digestion, you have your storage, and then you have your mobilization of your lipids. Okay, so firstly, you have in the small intestine, you have these large lipid droplets, right? Your emulsion droplets. And these are emulsified using bile, right? A hot word, emulsified, or broken down into these sort of things called micelles, okay? And that's facilitated by your bile salt, okay? Why does this process occur? Because it then gives your lipids a greater surface area for then your enzymes to act upon, which again, I guess Yajat touched upon as well. And there you have your enzyme, which is your pancreatic lipase, which then hydrolyzes the fats in micelles to produce, again, your fatty acids and your monoglycerides, which is just your monoglyceride, which is your glycerol. Yep, so one glyceride, glycerol. Okay, and the reason you sort of hydrolyze these micelles is that micelles cannot pass through the cell membrane freely. Okay, so then what happens, right? You're probably asking, then what happens? So then these free fatty acids that you have, and then this glycerol, because they can actually sort of pass through the cell membrane freely, they pass through the intestinal epithelium and move to the endoplasmic reticulum, or the ER, where again, they are resynthesized into your triacylglycerols, or your tags. And you're probably thinking, what is with this sort of assembly, disassembly, what's going on? And the reason your body does this is it's all to do with membrane permeability, right? Because it's important that it sort of assembles and disassembles it according to, what can actually pass through certain membranes. But again, I hope I've explained that pretty clearly. Just gonna have a quick look at the chat. Okay, no questions. And then you have your triacylglycerols, or your tags, and these are then packaged with cholesterol and phospholipids into these substances called chylomicrons. Really important term, comes up a billion times in exams, and it's really important to understand as well. So then you have these chylomicrons, and then they're exocytosed. Yep, and they're sort of put back into the venous system via these things called lacteals. Not too important, but I just thought I'd throw that in here. And then they are again broken down into, as they do with tags, into free fatty acids and glycerol in the blood, okay? Okay, so again, there's a lot of disassembly, a lot of assembly, but I hope I've explained that. Okay, so that's your digestion, right? So you've gone from that super fatty McDonald's Big Mac that you've had for lunch, and then I've sort of explained how it's gone from that Big Mac, how it's been broken down, and now your free fatty acids and your glycerol are now floating around in your blood, yeah? Okay, so that's digestion. Then we'll move on to storage. Okay, so you've got these fatty acids and you've got this glycerol that's sort of floating around in your blood. Then how does your body store this, right? Because we all have fat. Me, I've got a bit more than most people, but we've all got a bit of fat, right? So where is this fat stored? So you have two sort of areas of storage. Firstly, you have your adipose tissues. Important to note, your adipose only takes up your free fatty acids from the blood, not your glycerol, okay? However, it's able to synthesize glycerol from glucose, okay, which I'll go through in a bit more depth. So it's able to produce its own glycerol. It doesn't take it up from the blood, but it's able to produce its own glycerol, which it then uses to create triacylglycerols, your tags, which it uses for storage, okay? And then you have your second sort of side of storage, which is your liver, okay? So your liver, on the other hand, can take up your free fatty acids, and glycerol. Yeah, so that's a distinction that you have to make. And then it recomposes these two into tags for storage. Okay, all right. So we've gone through digestion, we've gone through storage, and that leaves mobilization. And this is a bit harder to understand, but again, I really want to make it as simple to understand as possible. And Bhuvi, I know that I've been talking for a while, so please let me know what you need me to hurry up as well. Okay, we've got half an hour. Let's do this. Okay, so your mobilization, what's important here? So you have your glucagon and your adrenaline, and they trigger the mobilization of your triacylglycerols, yeah? And so this entire process of mobilization is hormone regulated. Okay, so so again, like I've said, you have your two locations for storage, right? So you have your adipose tissues, and then you have your liver. Okay, so then we have to look at how do these two sort of areas, how do they mobilize these triacylglycerols? So in your adipose tissues, what happens is you have your hormone-sensitive lipase. This cleaves the triacylglycerol back into its, again, like I've said 1,000 times, into your 3 fatty acids and your glycerol, right? The fatty acids diffuse through the cell membrane and they bind to albumin in the blood, where they are carried to their various destinations, right? The heart, the skeletal muscles, the liver, etc., etc. And I think this diagram on the right really illustrates this pretty well. Yeah, so you have your hormones here that regulate the mobilization of these triacylglycerols. Here you can see you have your triglycerides, which is the same as your tags. You have your lipase here. It cleaves these into your fatty acids, yeah? And then this fatty acids enter your bloodstream, as you can see here. And again, they bind to this, this substance called albumin, okay? And then again, like I said, these fatty acids can travel to your heart, to your liver, to your muscle, yeah? But there's, in particular, I've left out the brain for a reason as well. And then you have your liver, yeah? So in your liver and intestines, what happens is your triacylglycerols are exocytosed into the blood, into the relevant lipoprotein, and lipoproteins I'll go through, but it's mostly your very low density lipoproteins, or VLDLs, yeah? So your free fatty acids that you have then, which are sort of transported, are either used to power the liver itself or are converted into ketones, and we'll cover this later. Okay, but just to go back to this diagram here, yeah? So in the liver, you have your glycerol, you have your fatty acids, as it's able to uptake both, yeah? And then your liver can make a decision to make your ketones, yeah? And the ketones specifically go to certain sort of areas of your body. So you have your heart, you have your brain, and then you have your muscle as well in some instances. And then you can have your triacylglycerol, which is sort of packaged into your very low density lipoproteins, and that can also be transported. Okay, so I hope I've sort of explained the mobilization of lipids in an easy to understand manner. But what I take away from this, one, is just understanding this diagram on the right, and two, just knowing that this is a hormone regulated process, and the two hormones that we like to sort of really look at are glucagon and your adrenaline, and there will be an MCQ question at the end that will concern this. Okay, moving on. So then you have your beta oxidation of these fatty acids, right? And you're probably asking, okay, so like, let's say I have that Big Mac, right? How come I feel super duper energized after I have that Big Mac? I can run a marathon. I can watch my lectures from week 2 in week 4. What is this energy that I'm getting, right, when I eat these really fatty things? Okay, let's explain this by looking at this image here on the right. Yeah, we love our images. Okay, so you have your long carbon chain here. And okay, but before I talk about it, one is that this process occurs in the mitochondria of cells. It occurs at rest. Think fatty acid, think lazy, think rest, yeah? It's a very high energy yield process, which is why after that Big Mac, you feel absolutely brilliant. And just to sort of really put this into context, for a 16 carbon chain, you approximately get 131 molecules of ATP, which is huge, right? So yeah, just to put it into context. Okay, so you have here your long carbon chain, yeah? And these are sort of broken down into 2 carbon units. And that process itself, breaking it into 2 carbon units, produces NADH and your FADH2, which are inputs into your electron transport chain, which gives you energy, okay? But not only that, what happens is your 2 carbon units are then converted into acetyl CoA, okay, which is your stage 2. And the acetyl CoA then goes through a citric acid cycle, like we've gone through, which produces ATP, okay, not a lot, but it produces some ATP, but it also produces your NADH and your FADH2, which again can go through the electron transport chain to give you a lot of energy. Okay, so here, the diagram here, you don't really need to know this, but here it sort of just explains how these 2 carbon units, yeah, your 2 carbon units, how they convert to your acetyl CoA, which is here, sort of that conversion. Okay, so then we have your glycerol metabolism, yeah? So I've talked about your fatty acids, but there's a key component of your tags that we still haven't touched on, and that's your glycerol. So what really happens to your glycerol? Again, not too high yield, but I think it's important. So what really you need to know is that your glycerol feeds into step 5 of glycolysis, okay? That's all you really need to know. So you have your glycerol here through a bunch of processes. It goes to your dihydroxyacetone phosphate, which I believe is a product of your step 5 of glycolysis. So it feeds into that. And then through glycolysis, it can be sort of made into pyruvate, okay? Which I'm sure I've gone through quite a few times and you all understand. Okay, quick check of the chat, nothing yet. Hope that's a good sign. Okay, so then you have your fatty acid synthesis, okay? So there's only really a couple things you need to know. Fatty acid synthesis occurs in the liver and your adipocytes. And the other important thing, the second thing you need to know is that the first step is a committed step, right? So it's when you go from your acetyl CoA to your malonyl CoA. Okay, so that step is the committed step, yeah? That can come up in the exam. They like to test that. But there's something else that will come up. And I hope at the end of it, in alcohol metabolism, I really want to tie everything together. But then you have your malonyl CoA here, right? And then that goes into your fatty acids. Okay, so you have your malonyl CoA, and what happens is that it elongates this malonyl CoA in a lot of steps, but it's the same thing. And it's by adding 2 carbon units. Yeah, so it adds a couple 2 carbon units, and that forms your fatty acid chain. The main thing to note is that this process uses NADPH, right, which we can consider the same as your NADH. And that will come up when you go through your alcohol metabolism and why do sort of chronic alcoholics, why do they get a fatty liver? And that's because your body favors this process of creating fatty acids through using your NADH that's produced when you metabolize alcohol. Okay, and then you have your triacyl, your tag or triacyl glycerol synthesis. So again, like I've said, your triacyl glycerols are your tags. They're stored in the liver and in your adipose tissue. Okay, so again, like I've explained a lot of times now, your adipose tissue is unable to uptake glycerol. So what it does is it actually produces glycerol. And so it first converts glucose, right? It has glucose. It converts it to through glycolysis to this product called glycerol 3-phosphate. And then, yeah, that's your glycerol, which you can then add to the fatty acids that it can uptake to produce your tags, okay? Lovely. And that, again, just to reiterate, your tag is your glycerol plus your 3 fatty acid chains. Again, this slide goes into a lot of depth on how it sort of produces this tag. But like I said, it's not really too high yield, and they're really not going to ask it at the end of the day. But again, I've included the information here if you do want to understand it a bit more. Okay, whereas in your liver, it can uptake glycerol, right? And it can uptake your fatty acids. However, to actually allow the sort of creation of your tag, your body requires the glycerol to be in this form of glycerol 3-phosphate. So what your liver does, it actually converts the glycerol that it can uptake it converts it to glycerol 3-phosphate, after which fatty acid chains are attached, resulting in your tags. Okay, lovely. Let's move on. So ketones, it's this word, right, that you've heard a couple of times. So you're probably like, what is ketones? Why does Noma keep mentioning it? I have no idea what's going on. I've walked into these breakout rooms on Zoom and I'm really confused. Yeah. So I aim to make this as crystal clear as possible for you. Okay. So you have your fatty acids. Yeah. Let's just say you have your fatty acids, and like I've mentioned, your fatty acids, your body likes to convert it to acetyl-CoA, yeah? And that acetyl-CoA then feeds into the citric acid cycle to produce your NADH and then your FADH2, which then goes into the electron transport change, la-di-da-di-da, right? That's how we sort of metabolize our lipids, okay? However, what can happen sometimes in your sort of state of fasting, right, is that there aren't enough intermediates in the citric acid cycle for that cycle to sort of rotate, right, and to produce that NADH and the FADH2. So what happens is, because there aren't these enough intermediates, it leads up to this buildup of acetyl-CoA, yeah? So the body sort of doesn't just want this acetyl-CoA to just remain like standing, right, and not doing anything. So what it does then, because especially since you're starving, right, your body needs this energy. So it can't just have acetyl-CoA lying there. So what it does is, in the liver, it converts this excess acetyl-CoA into ketones through this process called ketogenesis. No, good call. Really good question. I've just got a question in my direct message. So it's kind of asked why citric acid cycle intermediates deplete during starvation, and that's a really good question. And you know what? Let's figure this out together. Yeah. Okay. So let's go back in our slides. This is your citric acid cycle. Yeah. So for the citric acid cycle to go ahead, you need your NAD plus, right? And let's see, correct me if I'm wrong, but yeah, okay. So yeah, so suppose you have your food, right? You're eating. You're able to sort of go from using glycolysis, you're able to go from your glucose to your pyruvate, right? But then for your aerobic respiration, right, you need to go from your pyruvate to your acetyl-CoA, right? So your pyruvate decarboxylation, yeah? This process requires NAD plus as an input as well. So my answer to your question in my DMs, for whoever that is, would be that if you're starved, okay, you know what, I'll explain that in, let's see. Okay, correct me if I'm wrong, sorry. But when you're starving, you're not getting this glucose intake, right? So your glucose is low. And in glycolysis, I haven't included it here, but I'm pretty sure one of the inputs you need, or one of the products, is your NAD, or one of your inputs is your NAD plus as well. So if you're not eating enough and you're starved, you don't have your NAD plus, which when you go back to your citric acid cycle, if you don't have this NAD plus, then you're unable to sort of use it as an input. And then the citric acid cycle isn't able to function as it normally should. So whoever's in my DMs, I know that was a really shoddy explanation. I'm happy to come back to you after I'm done presenting, but does that kind of, in some shape or form, answer your question? You can just quickly shoot me a DM response. But I'm pretty sure it's to do with the fact that your intermediates or your NAD plus or your stuff that you're putting into the citric acid cycle for it to go around. And as you can see, there's a lot of things, right? They're not present. So whoever that is, if you could just quickly shoot me a Zoom DM, that would be good, and I can keep moving forward. So yeah, just read your response. So I think one of the intermediates that deplete could be your NAD+, but again, I'm not 100%. So I'll have a bit of a look as well through my own notes, and I'll probably get back to you. But all you really need to know in your ketones, right, is that your intermediates in your citric acid cycle, there aren't enough of them, which is why there's this buildup of acetyl-CoA, which is why then the body and the liver say, okay, we don't just want this acetyl-CoA to be lying around. How about we, actually use it to produce some energy, and then they convert this acetyl-CoA into ketones. However, there's another reason, which is really interesting, right? And sorry, I'm a bit too passionate for this time of the morning, but there are certain organs in your body, right, which only allow certain sort of molecules to pass through to allow it to sort of produce energy. And one of them is your brain, right? So your brain only likes glucose, and then it only likes ketones, right? If you're starving. So suppose if you're starving, right? You haven't got that glucose, right? So you're not eating, you're starving, you haven't got glucose. Then obviously, because it's your brain, right? Your body's in deep trouble if your brain doesn't have energy, right? Because your brain regulates the processes, your entire body. Your brain is essential to survive. So then to give energy to the brain, your body's sort of required to produce ketones, because it's one of those, it's the only second fuel source that your brain sort of can intake, which is again, another reason why your body sort of has to produce ketones. So again, just another reason. So another question people might ask is, why does the brain only like glucose and your ketones? And that's because your brain has this sort of area called the blood-brain barrier, and ketones are water soluble enough to cross this blood-brain barrier, and so is glucose. So it's the blood-brain barrier that sort of only allows ketones and blood glucose to go through. However, there's a flip side as well, right? You're probably thinking, okay, let's just produce these ketones and the brain's all happy. But ketones are also acidic. So if ketone levels are high in the blood, then what can happen is your pH can decrease, right? And that can also be pretty bad. And that's what we call ketoacidosis. Okay, so that's probably enough on ketones for one day. So I'll quickly go through cholesterol. Yajat went through this as well. But what's important, there's a bit of pathology sort of stuff. And pathology, by pathology, I mean kind of illness, yeah? So cholesterol, how is it synthesized, yeah? This synthesis occurs in the liver, the adrenal cortex, and your gonads, and it's synthesized from acetyl-CoA. So you can probably imagine how sort of interlinked all these processes in your body are, right? So what's important to note here, this sort of flow chart. Yeah, so we go from acetyl-CoA, we go to Hmg Coa, methylnoate, and then your cholesterol. So it's a sort of a three-step process. So the important thing to note is that your Hmg Coa converts to your methylnoate. So that's your sort of second step here. And it uses this enzyme called Hmg Coa Reductase, and it's a rate limiting step. Okay, so why is this process important? Because this process is self-regulating. So as Hmg Coa Reductase, it can regulate the concentration of your Hmg Coa. So in turn, by regulating the concentration of Hmg Coa, your body can actually regulate the amount of cholesterol it's sort of producing. Okay, so a bit of pathology stuff, yeah? So cholesterol, as you probably know, it's important, right? It's a precursor for bile salts. It's an important component of steroids, hormones, and vitamin D as well. And it's also a key to membrane fluidity. So it's important, but you've obviously as sort of year one med students, you've heard of cholesterol also having a few negative sort of impacts as well, right? Especially to do with sort of heart disease. So the reason I sort of talk about Hmg Coa Reductase and Hmg Coa and this entire process as being important is because it's important for your body to really regulate the amount of cholesterol it's producing. And if this process of producing cholesterol becomes unregulated, then that can really lead to an increase your likelihood of heart disease, and that's obviously really bad as well. So your body is really smart, and when you have illness, a lot of the time it's when this process of creating cholesterol is impaired. So you'll learn a lot about this in year 2 as well. But one thing to keep in mind is your Hmg Coa Reductase, Hmg Coa. That they're important steps. Hmg Coa Reductase is an enzyme, and it's rate limiting, and it's what facilitates the conversion of Hmg Coa to mevalonate. The reason I've sort of harped on about this for a while is because I have included a question on this. So remember these terms. Okay, then we'll be moving on to lipoproteins. Okay, we're a little short on time, so I'll try and run through this pretty quickly. So lipoproteins, yeah. What's important, and Gadget went through this as well, but very quickly, the hydrophilic on the outside and hydrophobic on the inside. There are 4 types as well, yeah? You have chylomicrons, VLDLs, you have LDLs, and you have HDLs. A thing that faculty likes to talk about is that LDLs are considered your sort of bad lipoproteins. Why? Because it mostly, it transports cholesterol to tissues, yeah? So you consider this as sort of being your bad lipoprotein. You can conversely, you have your HDLs, which are considered so your good lipoproteins. And why? Because they mostly transport cholesterol away from your body's tissues, which is good. VLDLs, they're synthesized in the liver, and they transport your triacylglycerols, your tags, from the liver to other tissues between meals. And I've talked about this, in the slides prior as well. Then you have your chylomicrons. They're synthesized in the intestinal cells, and they transport your dietary tags into your blood. Yeah? Okay, lovely. Alcohol metabolism. This is an important area because the faculty likes to test it, and also because it really ties together a lot of what we've been talking about. Okay, so the first sort of pathway I'll be talking about is your normal metabolism of alcohol. And I'd like to start off with a bit of sort of real life example, right? So I'm sure you've had a night out and you've seen a friend who's been a bit red in the face, right, after consuming maybe a few beers. And the reason for that is, and we call this phenomenon Asian flush. And why do people get Asian flush? Because there's this buildup of this substance called acetaldehyde. Yeah, so the next time your friend is a bit red, let him know that he's got a buildup of a bit of acetaldehyde. Okay, so let's go through. There's a lot on this slide, but I'd really like to go through some of the important stuff. So as you can see, to metabolize your alcohol, one of the inputs you require is your NAD+. Yeah, this is really important. One of the inputs for aerobic respiration is your NAD+. Yeah. So since your alcohol metabolism requires NAD+, and your aerobic respiration requires NAD+, what your body says is aerobic respiration is then less preferable. So then what it uses is your liver cells use anaerobic respiration to produce NAD+, okay? So that it can use the NAD+ that it's created to then metabolize your alcohol, okay? To allow this metabolism to continue. Does that make sense? So your liver cells use anaerobic respiration to produce NAD+, so that alcohol metabolism can take place. Okay, first point. Then what happens is, what I've sort of talked about in the past is anaerobic respiration, as I've said, is less energy producing, right? So your body isn't producing as much energy. So you get fatigued, yeah, as your body's trying to metabolize this alcohol. And that's what we call hypoglycemia, okay? And because you don't have the energy, right? You don't have the energy when you have, when you do your sort of anaerobic respiration. You also don't have the energy to convert that lactate, right? When you convert your pyruvate to your lactate in anaerobic respiration, you now don't even have the energy, the ATP, to go from lactate to glucose, right? Gluconeogenesis. Do you remember that left-hand side column that I was talking about? A heap of slides before. So your body doesn't have the energy to convert that lactate to glucose, which is your gluconeogenesis. So what happens is you have this buildup of lactate, or the production of lactate from anaerobic respiration, and that causes this phenomenon called lactic acidosis, which is not good. But again, what happens is alcohol metabolism, one of the outputs is NADH, right? And in the production of fatty acids, right, remember that sort of committed step when you go to malonyl CoA and all of that? Yeah, you go acetyl CoA, malonyl CoA, and then you add the two carbon units each time to produce your free fatty acids. That process, right, actually uses NADH. Or yeah, so when you metabolize alcohol, you have this buildup of NADH as well. That sort of which is an output, right? So then what your body does is it uses this NADH that it has to produce fatty acids. So now what's happening is you've got all this fatty acids sort of accumulating in your liver. And that is what we call hyperlipidemia, which is, the phenomenon of chronic alcoholics having fatty livers. Okay. All right. This isn't really important, so I'll really quickly go through this. It's 11:55. So your microsomal ethanol oxidizing system. This is used when there's a heap of alcohol. Doesn't really come up, not too important in my opinion. But again, I've included the information here. You can look through this in your own time. Okay, I'll really quickly go through amino acids, okay? Because I want to go through some questions because I think you'll learn and really you'll apply the knowledge that you've hopefully accumulated through this revision lecture. Okay, amino acid overview. Amino acids are either taken in by diet or broken down from muscles, and they're catabolized in the liver. Amino acids are used to synthesize glucose and acetyl CoA. Yeah, that's all you really need to know. This diagram here is sort of your amine group, your carboxyl group, and then your variable group. I'm 99% sure this is covered in 3-4 Chem as well. So hopefully being year ones, you remember this as well. Okay, so a few processes that I'll be going through in really quickly, right? You have transamination. What is this? It's the synthesis. It's important in the synthesis of non-essential amino acids. So your essential amino acids are taken in by diet only, whilst non-essential can be created in the body from other amino acids. What happens in really simple terms? The amino acids and the keto group, yeah, they swap. They're keto and amine group. Yeah, that's pretty much all you really need to know of transamination. Then you have deamination, yeah? So deamination is the elimination of an amino group, sort of in the name, from an amino acid, forming ammonia, yeah? Leaving a carbon skeleton that can be respired for energy, okay? That's all I'd really take from this as well. So it forms ammonia and also leaves behind a carbon skeleton. Then you have your urea cycle. So what feeds into it? So why do you have your urea cycle? It serves to detoxify and make your water soluble. So yeah, pretty much it serves to sort of excrete the ammonia produced in deamination so that it can be excreted, and this process takes place in the liver. Again, not really too high yield, but again, the information's here. Then you have your glucose alanine cycle. Draws a bit of parallels with the Cori cycle that I've gone through. So pretty much like I've talked about, in starvation, some amino acids can be transaminated in muscle cells to alanine, yeah? So you have your amino acid, it goes into alanine, yeah? This amino acid is transported through the blood to the liver, yeah? So this column here is the liver, where it's broken down to pyruvate. The pyruvate is then used in gluconeogenesis. Yeah, it has an input of ATP to become glucose, right? So that pretty much explains how you can use your muscle to breakdown of muscle to produce energy. However, what can also, a bit of this sort of alanine sort of goes to your ammonia, and then your ammonia goes into your urea cycle. Again, very quickly, amino acids, not too high yield. Okay, energy sources of cells. I've talked about this quite a bit throughout my presentation, but let me quickly go through what are the important points. Cardiac muscle, yeah. So you have your heart. What is your primary sources of energy for your heart? The main one is your fatty acids, okay. But when fatty acids aren't available, it's glucose and ketones. That's all you really need to know about your heart, yeah, and the heart sort of cells. Then you have your brain. I've said as well, your brain primarily uses blood glucose, but if you're fasting, it can use ketones. Your red blood cells only respire glucose because they don't have organelles, so they don't have mitochondria as a result, so they can't do aerobic respiration. So thus, your red blood cells only respire glucose using anaerobic respiration. And then you have your skeletal muscle. Sorry, I just got a message in my chat. Okay, feel free to run over time. Thanks, Spoovy. Okay, so you have your skeletal muscles. At rest, like I've said, you think rest, sort of fatty acids. So at rest, your skeletal muscles use fatty acids. This will be really important in a few slides. If you need a burst of energy, it uses creatine phosphate. Yeah, you really don't need to know this in a lot of depth. Just know at the start of a sprint, at the end of a sprint, yeah, in the first bit of any sort of real bit of exercise for your skeletal muscles, it uses this substance called creatine phosphate. Moderate activity uses glucose, using your glycogen stores, then fatty acids and ketones as well. And for sustained activity, it uses your lipids or your triacylglycerols or your tags. Okay, something really high yield, yeah? What's really unfortunate about metabolism is that we've gone through carbohydrates, we've gone through lipids, we've gone through our amino acids, but unfortunately faculty really doesn't like to ask many questions on these macromolecules. What it really likes to ask questions about is the great metabolic race, which kind of puts everything together, okay? So this slide here, what I'd recommend is really taking a screenshot of this, inserting into your notes. It's really vital because before the exam, if you just have a read of this, you're more than sort of prepared to answer any questions in the great metabolic race. And like I've said, great metabolic race is what they really like to assess. Okay, what's important here is you read through this table and you have a bit of an understanding. At rest or before the race, our skeletal muscles, they like to use fatty acids. In the first couple of minutes, right, or in the first 2 minutes, you're probably thinking, what is with this anaerobic respiration of stored glycogen, right? Why is it anaerobic respiration? And the reason for that is your skeletal muscles are sort of getting used to this sudden increase in exercise, right? So then there's a bit of anaerobic respiration. However, after that, it goes back to aerobic respiration. Another important note is that at the 30-minute mark, there is this intersection of the use of fats and carbohydrates. And again, another thing to note is at the final sprint and at the start as well, your skeletal muscle cells use creatine phosphate. Okay, and again, like I've said here, is you have your proteins, right? So you're thinking 8 hours. So at 8 hours, you've probably run out of your fats, your glycogen, or your carbohydrates, and you're starving, right? You're at rock bottom. Then all other energy sources have been depleted. So your muscles, right? Or your muscles, your protein, they're broken down into amino acids. Your amino acids, like I went through a couple of slides ago, are then used to, you go from your amino acids to alanine to pyruvate to glucose, yeah, using gluconeogenesis. So then you use your amino acids. After 8 hours, hopefully no one's going on a run for that long, but then your body breaks down your muscles. Another sort of real world example of this is, not that you guys have watched documentaries, but I'm sure maybe one of you, have you watched those documentaries of this guy? He's stranded at sea in the Pacific Ocean for 60 days, right? And you've probably looked at the faces of those people, right? And they're hollow, their limbs are just the bone. They've lost pretty much all muscle mass, right? And what's the reason for that, right? So they're starving. Yeah, they've got no real input of carbohydrates. They've utilized all the fat that they have in their body. Then the body, right, the process of using your proteins, using amino acids, using that muscle kicks in, which is why those people look so hollow and they look so, they just look so deprived of muscle, of muscle mass. Again, a real world example to put that into context. Okay, another thing to note here is that very early on in the great metabolic race is that there's a use of fat, for the first, let's say 3 minutes. There's a use of fat, after which your carbohydrates sort of spike, your fat levels, your fat as a percentage of energy supply decreases, and then the fat as a percentage of energy supplies sort of increases, and your carbohydrate as a percentage of energy supplies supplied plateaus. Okay. Again, like I said, great metabolic rate is super important. Okay, so now I've harped on for way too long. So we'll be going to some questions. Please put your answers in the chat for two reasons. One is that, yeah, you can be a bit more engaged. It's a time for you to sort of, test your own knowledge. But the second reason is it also gives me a bit of an indication of whether this revision lecture has been helpful, whether you've taken anything from it. So it'd be lovely to see all of you absolutely smash all these questions. So what I'll do is, again, this is a safe space. If you get a question wrong, no one will, this is a no judgment area. So you can put your answer in and it's completely fine, even if it's wrong. So I just encourage everyone to put their answer in so we can, go through as many questions as possible. So I think there's 19 questions. Okay, so first question, Pranay is a state sprinter. Yeah. When he starts the sprint, what energy source is used? So if possible, put your answer options, just type in A, B, C, or D into the chat. Feel free to unmute yourself as well and yell out the answer if that works well. Eric or Bhuvan, you guys can yell out the answer if no one else. D, perfecto. That's a good sign. Everyone seems to be listening. D is the correct answer. Okay, moving on. It has been 4 hours since Kiran had her last meal. Which processes are currently occurring in her body? There are more than one answer. Bit of a tougher question. So when giving your answer, you can be like B, C, F, or A, B, C, that sort of thing. And Eric, I'll probably run for 5 more minutes. So okay, all right, a few mixed answers. So with a lot of these questions, you'll find there are often quite a few sort of disputed answers. The answer in this case is actually B, C, and F. Okay, let's have a go. So the reason we say there is no glycogenolysis, is because by 4 hours, chances are the glycogen that your body has stored are probably depleted as well. So I hope that makes sense and why A. I think most of you have put C and have put F. So yeah, and I think Susan put B, so gluconeogenesis as well. So at 4 hours, your body is probably pretty starved as well. So then, like I've said, in a starved state, the body looks to go to the process of gluconeogenesis. Okay, all right, few more questions. Question 3, choose the statement which is true. So just put in your answer, A, B, C, or D. A. You guys got the right answer. Perfect. All right, question 4. Which of the following statements is false regarding the Cori cycle? Sorry guys, had to do it. Cori cycle, we didn't go through it in a lot of depth, but hopefully you can get this right. Yeah, Justin, really well done. So pyruvate conversion into lactate occurs in the bloodstream. Really, really good. Happy. So for those who didn't answer, was it sort of not well explained? Or were you sort of just looked at Justin's answer and you're like, yeah, he's got it? I know it's kind of hard to answer that question, but yeah, Justin, like we'll move on, but Justin is right. So pyruvate conversion into lactate does not occur in the bloodstream. Yeah. Okay. Question 5, which of the following is true? So do you guys, yeah, okay. I'm very happy, yeah. So I sort of told you guys that Hmg Coa Reductase, it's a rate limiting step. Very important when you look at sort of the synthesis of cholesterol. Cholesterol plays a huge role in a lot of sort of illnesses and pathologies that we see. So Hmg Coa Reductase is a huge and really important thing to take note of. Okay, which lipoprotein is mainly composed of cholesterol? This is a little harder. Actually, so these are all, okay. I think Susan's got it. Yeah, the answer is A. And then question 7. So if you guys remember, there's a committed step when you sort of synthesize. Nearly gave the answer away. Yeah, okay. So yeah, like I was saying, the committed step of fatty acid synthesis, you go from acetyl-CoA, malonyl-CoA, and then you have your in 2 carbon units, you sort of put them all together to form your fatty acid. Lovely. Question 8, which hormones act on receptors to break down tags? There's more than one answer. So do you remember the mobilization of your lipids, or your mobilization of your triacylglycerols? A and C, lovely. Question 9, past exam question. Starch and glycogen, which are both polysaccharides, differ structurally in what? Glycogen is what, whereas starch is what? Yep, you're right. Yep, Pranad, you're right as well. Answer is A. The next one, this has confused a few people in past exams, so have a good read. Which of the following pairs of statements correctly describes the differences between mobilization and digestion, of carbohydrates? Correctly. So just to refresh, your glucose 6-phosphate is a part of your glycolysis. Yeah, Susan, you're right. It is B. Good. It's a bit of a tougher one. So for those who found it tough, yeah, the slides are here. So yeah, have a go at this question again. Yeah, it's one of the tough ones for sure. Question 11, not too many. We're gonna really speed through. We've got 3 minutes left. So why is urea production increased during starvation? Yeah, so we talked about yeah, good. Thanks, Justin. Really good. So sorry, just for those who didn't catch it, the answer for that was B. And in the speaker notes, you can see the answers as well. Question 12, if you have consumed a meal containing carbohydrates, protein, and fat, which of the following would be transporting the dietary lipids in the blood? Let's go, guys. Smash it out. Dietary lipids. So what's that first sort of transport molecule that we see? E, yeah, chylomicrons, good. Monash lecturer has been sitting at her desk for 4 hours. Mrs. Lunch is not eating for some hours. Which of the following metabolic processes would be active in her cells after such a 6-hour fast? Sorry, this is probably really badly typed, or the question itself was a bit wrong. Yeah. Yeah, no, this is an interesting question. There was a little bit of conflict in our year because we had this past question, and there was a dispute between sort of answer options B and C, because it is a bit arbitrary between like what hour does your body, like do your glycogen stores finish, and at which stage do sort of your fatty acids kick in. So it's tough. So I'd probably pay answer option B and C. However, what faculty said was C. That's the answer option they backed. A runner is about to bring a long run, standing at the starting line. So he hasn't started his run. What's the sort of fuel source? So prior to his run. Yeah. So remember, prior to a run, you're using those fatty acids. Yeah, you're at rest. Okay, you know what, in the interest of time, let's go through. So 2 molecules of glucose underwent glycolysis to create 4 molecules of pyruvate. How much ATP is generated from this process? So B, good. So you guys understand it, you know the ratio. If blood glucose levels are low, 2 more questions, Prudy and Eric. If blood glucose levels are low, which of the following yeah, so if you could, guys, just type in an answer to this question. Yep, perfect. The answer is A. And last question, lucky last, the finale. So Hazza comes in the hospital, night of drinking, complaining of nausea, vomiting. Doctors determine excessive amounts of acetaldehyde in his blood, the thing that causes Asian flush. Which of the following is likely cause of this issue? B, lovely. So again, I didn't cover this in too much depth, but there is an enzyme that allows the conversion of your ethanol to your acetaldehyde. And that enzyme is your acetaldehyde dehydrogenase. So that's why he's probably deficient in that. I think it's actually when you go from your acetaldehyde. Sorry, I'll literally finish on this one, guys. I'll go back just to explain this. Because they might ask this. Okay, yeah. So so when you go from your acetaldehyde to your acetate, you use this enzyme called aldehyde dehydrogenase, or acetaldehyde dehydrogenase. So what happens is, to have a buildup of acetaldehyde, you can either have a deficiency in this enzyme, or you can have this enzyme working like in double time. Yeah. So if this enzyme works in double time, which is your alcohol dehydrogenase, that can also lead to a buildup of this, because sort of when you look at the ratio of sort of the rate of reaction, if this reaction is happening faster, it will also result in a buildup of this. And then conversely, if you have to go from here to here, you have this sort of enzyme not working correctly, that can also lead to a buildup of acetaldehyde. Anyway, that concludes my presentation. So that's a lot of slides. I think it was close to 80 slides. Thanks so much, everyone, for tuning in. Hope you learned something from that. Again, it is an early morning on a Saturday after a big week. So really appreciate everyone who came in, everyone who contributed, and even to those who were just listening, really appreciate it. You can my contact details are on the first slide. But other than that, take it away, Eric, and take it away, Prudy. Thanks so much.
Huge thanks to Yash. That was an amazing revision lecture. Thanks so much, Yash, especially for battling through with your voice. But yeah, amazing. All right. I guess if everyone's okay with after questions, we'll probably jump straight into Eric's revision lecture. So take it away.
Easy, I'll get started. Yash, that was great for you, buddy. That was a lot of content. I couldn't have done it myself. All right, let's get started then, guys. Mine's a bit easier. Mine's just on genetics. Way easier than a lot of that mobilization of fats and metabolism and all that sort of stuff. So we're cracking. Really should be short, shouldn't be too long. So yeah, let's get started. So genetics, I just put a credit slide there. So what am I going over today? Central dogma. I'm just going to pin my screen 1 sec. Central dogma, DNA replication, transcription, translation, patterns of inheritance, genetic mutations, population genetics, and biotechnology. In general, I think it's quite comfortable in terms of the content, especially if you've done things like VC biology. A lot of this should be quite useful. Like, you probably would have seen this stuff before. Okay. I guess I haven't introduced myself yet, but if you don't know me, my name's Eric. So yeah, let's get started. So first one, DNA. What the hell is DNA? So DNA is deoxyribonucleic acid. So it's one of the most important molecules you're going to need to know. It's a polymer. It's made up of subunits called nucleotides. Okay, so how do we actually get this DNA thing? It's made from condensation polymerization, and it's made by covalent linkage bonds between sugar and phosphate. And that's why you get something called a sugar phosphate backbone. Okay, and then another thing you need to know is that it's a double-stranded helix. And it's in an anti-parallel arrangement. So it goes from 3' to 5' on one end and 5' to 3' on the other end. Okay, so those are the basic information you need to know for DNA. And I'll get to why a lot of this stuff that I've just mentioned is quite important. Okay, good. So what are the important other kind of features you need to know? So we talked about double-stranded uniform diameter. It's a right-handed twist. I think that was quite something that I found quite interesting. So it goes like right-hand twist, yeah. Has nitrogenous bases pointing towards the center, and then it's got your hydrogen bonds giving you some complementary bases in the middle. So one of the key things to remember is that adenine always binds with thymine, okay, unless it's RNA for uracil, and guanine binds with cytosine, and it has 3 hydrogen bonds. So 2 hydrogen bonds versus 3 hydrogen bonds. The way you remember this, or the way I remember it, is that G and C rhyme with 3. Okay, so GC3. So that's how you get, remember, guanine and cytosine 3 hydrogen bonds, whereas adenine and thymine are 2 hydrogen bonds. Okay, and you can see the arrangement of what DNA might look like. One going from 5' to 3', and the other going from 5' to 3'. So they're anti-parallel arrangements. Good. What are the 4 essential functions of DNA? So this is something I remember they stressed in a workshop or something. So it's also quite important to remember. So there's 4 things. It stores your genetic information, or how you make your proteins and that sort of stuff. It reads and allows for gene expression. It's susceptible to mutation. So in general, mutation is kind of perceived as a bad thing, but mutation is actually good. And I'll talk about why it's good. But mainly because mutation, it can allow you to make new sorts of proteins. And that's very good for things like evolution. And genetic diversity. Okay, and 4, it must be precisely copied. It's a bit weird. I'm not too sure about these 4 sort of essential functions of DNA, but I remember these are the 4 that they stressed. So please remember these 4. Good. What's the difference between chromatin and chromosome? So chromatin is the unraveled structure of DNA. So it's just like a long chain of thingo, whereas chromosome is your condensed form of that. So you would have maybe heard these terms before, chromatin and chromosome. So I'm just giving you a bit of a definition there. So if you look at a diagram, chromosomes are really condensed form DNA, whereas chromatin is that uncondensed form DNA. Okay. Now, you would have seen here, there's these little things called nucleosomes, okay, which make up your chromatin fiber. So what is a nucleosome? A nucleosome is the fundamental subunit of the chromatin. And each of these nucleosomes are made up of 8 proteins called histones. Okay, and that's why you get something called a histone octamer. Now, these proteins, I remember briefly, there's 4 types of these proteins. I don't remember what the 4 are called, so it's not too high yield. I'm going to be going through some of the high yield content today, stuff that you really need to know. Okay, so stuff like that, the specific little details, I wouldn't worry about. These nucleosomes are then what we call formed highly and organized, and then you condense it, and then you get something called a chromosome. Okay, and I'll talk about chromosomes later on, but you've got 46 of these chromosomes in a normal human cell. Okay, good. So what's the difference between a purine and a pyrimidine then? This is easy stuff. So A and G, or adenine and guanine, are purines. Okay, the way to remember it is that first letter of A, T, G, and C. Okay, so A, T, and G, C. A and G are your purines. Purine is the largest two-ringed molecule, nucleotide, whereas pyrimidine is your single-ringed molecule. Now, this is important because a purine always binds with a pyrimidine, and that gives you 3 rings. Okay, that's its functional importance. Uracil is also the other pyrimidine, but uracil is only found in RNA. Okay. All right, that's a lot of the bulk about little details you need to know about DNA and little things about its background and all that sort of stuff. What about DNA replication? How do you actually replicate DNA? So I've attached a video here. I'm going to watch this video with you guys. This is probably one that after you watch it, everything just makes much more sense. Okay, but we'll speed up to like 2 times speed, so it should be all right. Let me know if the volume and whatnot isn't working, but in general, it should be okay. All right.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape.
Ooh. Let me try that again.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continually, the DNA polymerase adding bases 1 by 1 in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, so that's pretty much all you need to know from DNA replication. I wouldn't say it's, when you read it at the start, it's kind of a little bit difficult because you don't know where things are and whatnot. I mentioned a couple of things. So what are the roles of topoisomerase and helicase? So you would have seen helicase was the first enzyme that actually unzipped it. But there's actually something called topoisomerase, or I don't know how you pronounce it, but that's actually the thing that actually unwinds DNA before DNA helicase. So both of these actually unwind DNA. It's important to know these 2 enzymes because later on you guys will be learning about things that actually stop DNA replication and whatnot. So you can have things that actually block these enzymes, topoisomerase or DNA helicase, and therefore you stop transcription and the production of proteins. Okay, so topoisomerase unwinds the DNA, and that allows DNA helicase to then unzip. DNA helicase will then allow for the local unwinding and produce that original replication, or that replication fork that we talked about in that video. Okay. So what is the role of RNA primase? So we would have seen RNA primase was the thing that actually is producing those RNA primers, and then that allows the DNA polymerase to find those RNA primers, and then for the DNA polymerase to then copy that DNA. So RNA primase produces these RNA primers, And then these RNA primers are later removed. Okay. So what about DNA polymerase? What was the role of that thing? So DNA polymerase was a thing that binds the RNA polymerase. Sorry, yeah, binds to the RNA primer. And it allows for the catalyzation of these DNA nucleotides from a 5' to 3' direction. So it goes in a complementary base pairing. So always A binds with T and G binds with C. And then it pretty much produces that extra strand, okay, on both the leading and lagging strand. So something to note is that once you add these nucleotides, they're actually something called nucleoside triphosphate. So it's actually made up of, if you look at the diagram here, it's made up of a, you got your deoxyribose, but it's got 3 phosphates. And then what actually happens is that 2 of the phosphates actually drop off, and then you get that bonding into that DNA. And when you break off 2 bonds, that actually gives you the energy for this process to actually occur. Okay, which way is the direction of DNA synthesis? DNA synthesis is always from 5' to 3', but it's red from 3' to 5'. So please note that. I think it's very confusing to know which way is which. Just remember that it's 5' to 3' is the direction that the DNA polymerase is running, but at the same time, the DNA polymerase is reading at 3' to 5'. Now, what am I talking about in terms of 5' to 3'? 5' to 3' comes from the order of the carbons. So if you guys have done chemistry before, you see carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So the direction of 5' to 3', let's talk about the direction there, okay? Now, what was the leading strand? So the leading strand was the one that just goes quite straightforward, pretty much. The DNA polymerase is able to grow that new strand, the semi-conservative strand, from a 5' to 3' direction until that's complete. And then the RNA primer will then be degraded and replaces it. So this is the leading strand here. And it's the one that's quite smooth. The lagging strand is the one that needs to go back and forth, back and forth, back and forth. And I'll talk about how it does that. So the lagging strand was the one that grows in the opposite direction. So you know how I told you guys that DNA polymerase can only go from 5' to 3' direction, right? So because the strand is going in the opposite direction, it has to just go back and forth, back and forth, back and forth in order to fulfill that, making that whole one strand. So these back and forth movements are called Okazaki fragments. And these are made, these Okazaki fragments is what allows the lagging strand to be able to produce that new strand of DNA. So the DNA polymerase must be able to skip past these 5' ends. And then the DNA ligase, which is kind of like a glue, okay, that kind of glue is able to stick these gaps between these Okazaki fragments together to then get you a new semi-conservative strand. So the leading strand is the one that is able to just give you a straight strand, new DNA strand, whereas the lagging strand has to go back and forth, back and forth. Okay, and that's pretty much all you need to know for DNA replication. If you guys are familiar with the process, that first video, they'll get you the majority of the marks, okay? Yeah, that's pretty much it for DNA replication. So what about the DNA repair system? How does DNA actually repair itself? Firstly, you guys need to know about the difference between telomeres and telomerases. If you guys have heard Craig Hassett already, Craig loves to talk about telomeres. So telomeres are the endings of chromosomes. And what normally happens is that they normally get shorter and shorter, especially if the DNA is getting damaged. So how does it actually get shorter and shorter? So the leading strand actually precedes the end, so that the lagging strand has a 3' overhang or a terminal gap. So there's a gap each time you do DNA replication, and therefore causes the chromosome to get shorter and shorter after every time. The only way to fix that is by something called telomerases. So telomerases are these things, ASEs, if you guys have heard of enzymes. Enzymes always end in ASE. So telomerase is responsible for telomere synthesis. So what it does is that it's got something called RNA template, and that allows the DNA polymerase to add on these extra nucleotides and therefore remove that overhang. Okay. What are the DNA proofreading and repair systems then? There's 3 that you guys need to know. And these 3, they always test in exams, so I'll take quite a big note on this one. So proofreading, mismatch repair, and excision repair. So proofreading, the way I think about it is that like you proofread during your exam. So proofreading occurs during the replication process. Mismatch repair, so the teacher checks for mismatches after your exam. So a mismatch repair scans and repairs errors after replication. And an excision repair, excision is to kill, so it happens after or throughout life. So an excision repair mechanism operates over the lifetime of the cell, and that results in from like things like chemical or radiation damage. Okay. So please remember these 3. If you have your own way of remembering it, then that's fine. Just remember that proofreading is during, mismatch is after, and excision is throughout. Okay. Now, transcription is easy stuff, especially if you've done VCE biology. Okay. So in terms of how we go from DNA to protein, RNA is, so for what transcription is when we produce RNA or RNA transcript in the nucleus from the DNA code. Okay. That RNA will then be modified and turned into something called mature mRNA, and then leave the nucleus and enter the cytoplasm to attach to the ribosome, either in the cytosol or the rough endoplasmic reticulum. The protein is then made by the ribosome based on the mRNA code, and then amino acids are brought to the ribosome by tRNAs. That's called translation. Okay, so that's the difference between transcription and translation. So let me delve into transcription and what's happening here. So in transcription, you've got a big thing called RNA polymerase. Now, this big enzyme does a couple of things. It unwinds the DNA, and then it also creates this pre-mRNA strand by complementary base pairing of the template strand. And then this pre-mRNA strand will pretty much match up to your coding strand, except thymine is replaced by uracil. Okay. So step 1, RNA polymerase attaches to the DNA 5' upstream of the protein coding region. Okay. And then the double-stranded DNA of the gene unwinds, and that exposes the bases of the template strand. So once you've exposed the bases of the template strand, there will be complementary base pairing of the RNA nucleotides against the template strand, and therefore you produce something called an RNA molecule, or specifically a pre-mRNA molecule, and then that will be continuously transcribed. So as the RNA polymerase moves along that DNA template, more RNA nucleotides are joined to that ever-growing RNA molecule. So you get a longer and longer pre-mRNA molecule. And then once transcription is stopped, the RNA polymerase will detach from the DNA, and then you get a single-stranded pre-mRNA molecule. So this pre-mRNA molecule is complementary. So complementary meaning if it was an adenine, then it'll be a thymine. Okay. It's complementary to the template strand, but it's exactly the same as the coding strand, except thymine has been replaced by uracil. Okay. And that's pretty much transcription. Okay. That's pretty much all you need to know. After transcription, you got something called post-transcriptional modification. So in order to get from pre-mRNA to mature mRNA, there's 4 things that has to be done. Introns are removed, and then exons are spliced together, and that is by the action of a spliceosome. Now, I'll highlight this word splice. You have to use it correctly, because a lot of students actually use it splice as in like slice. So they think that when you remove introns, it's slicing them out. Except spliced actually means joining together. So you guys got to be careful about that. So the introns are removed, and then the exons are spliced together, meaning they're joined together. There's something called alternative splicing. Alternative splicing is when you shuffle around those exons, okay? And that gives you a new pre-mRNA, new mRNA strand, and that gives you a new protein because you've shuffled around those exons, okay? Two other things, a methyl cap is added on the 5' end, and a poly A tail is added to the 3' end. The reasoning behind why we add a methyl cap and a poly A tail is because the cytosol is actually quite toxic. So because the cytosol is quite toxic, what we want to do is that we want to protect that mRNA from being damaged. So in order to do that, we add a cap and a tail. Okay. So the mature mRNA will then leave the nuclear pore, okay, which is obviously on that nucleus, and travel to a ribosome. That ribosome, whether that be on the rough endoplasmic reticulum or in the free-floating cytosol. Okay. So just sorry, I've just added here. This is the spliceosome. The spliceosome is made up of these things called SNRPs or SNRPs. And what they do, sorry, I've lost my voice. Oh, sorry about that. What I was trying to say was that these SNRPs form those spliceosome, and that's able to remove those introns, and then you get these exons left. Okay. The way to remember whether introns are removed or exons are removed is that introns, they start with in, so it's like they interrupt the sequence, whereas exons are expressed. Okay, so in for interrupt, X for expressed. Okay, so introns are removed, exons are spliced together. Please remember that. Good. What about translation? So after we've done transcription, we've made our mature mRNA, and that mature mRNA would then go to the ribosome to do translation. So this is how we're going to actually get our polypeptide chain. So right now we've done our DNA, we've produced that mRNA, okay, in the nucleus, and that's now the mRNA has moved out of that nuclear pore to a ribosome. So what actually happens here? Let me explain. So there's kind of four so the mature mRNA will enter the cytoplasm, leaving the nucleus by the pores in the nuclear membrane. So they know that ribosomes aren't actually just a single thing. They're actually two subunits. So there's a large subunit and a small subunit. These ribosomes actually then assemble together around that mRNA, and that actually allows for translation to occur. So tRNA, or something called transfer RNA, which are these molecules I've pictured here. Okay, they look like 3-leaf clover kind of things. What they do is that they carry these amino acids to the ribosome, and they match their anticodon to the codons on the mRNA to then drop off that amino acid and to create those peptide bonds, and that gives you your polypeptide chain. So you look here, the mRNA is read in a 5' to 3' direction in codons, and it finds its matching anticodon in tRNA, and therefore it drops off those amino acids to get your polypeptide chain. Okay, that's translation, and it's easy as that. Okay. What about patterns of inheritance then? These are things that have to do with genes. So after we've done all that central dogma stuff, so we've done all what DNA, we've done transcription, DNA replication, translation. How is this relevant? So what is a human karyotype? You would have heard this phrase before. So a karyotype is a pattern or a picture of chromosomes grouped by pairs and organized by size. So humans have 46 chromosomes in their cells. We have 22 pairs of autosomal chromosomes and 1 sex chromosome pair. So males have 22 autosomal pairs, an XY sex pair, and females have a 22 autosomal pairs, an XX sex pair. Okay. So when you look at a human karyotype, you might see something like this, where the chromosomes are matched up and paired by size. Okay. So the main thing to know is that males are XY, whereas females are XX. Okay. Now, there are some important genetic terms you guys need to note. So there's a trait, phenotype, genotype, gene, allele, locus, homozygote, and heterozygote. So a trait is a characteristic. So it's kind of like a trait might be like your hair color or something like that. A phenotype is a physical expression of that gene. So the phenotype might be like black hair. The genotype is the genes that actually code for that phenotype. A gene is a section of DNA. An allele is that variant of a DNA. A locus is the location of a gene on a chromosome. Homozygote is when you have two copies of the same allele, and heterozygote is when you have two different alleles of the same gene. Okay. Become familiar with these terms because these terms are always going to be mentioned. And I think it's actually relatively quite easy. Okay. So just make sure you guys know that. Now, what about dominant and recessive traits versus codominance? What are these talking about? So dominant traits is when alleles produce the same phenotype, whether it's paired allele or is identical or different. So whenever you have you would have seen like big A, or big A, little a, okay? If that's expressed as a trait, then it's called a dominant trait. Whereas recessive traits, you need to be homozygous in order to be expressed. Typically, it's going to be like little a, okay? Whereas codominance is something that's quite interesting. It's when both alleles are simultaneously expressed. So these are things like the ABO blood group. Okay. What about autosomal dominant and autosomal recessive? So autosomal inheritance, firstly, is when the gene is located on one of the autosomes. Okay, that means that it's not on a sex chromosome. So this means that males and females are going to be equally likely to inherit that gene. So autosomal dominant is when one mutated copy of the gene in each cell is sufficient for a person to be affected by an autosomal dominant disorder. And the one that you guys need to know is Huntington's disease. Okay. Autosomal recessive is when both copies of the gene in each cell have mutations, and then the parents of an individual with an autosomal recessive condition each carry a copy of that mutated gene. And the one that you guys need to know is cystic fibrosis. Okay. You might come across these tables here. And in order to get better at reading these, all you need to do is just practice them. Okay. So the one on the left is autosomal dominant, and the one on the right is autosomal recessive. What about sex-linked then? What about sex-linked inheritance? What is that talking about? What's the difference between X-linked dominant and X-linked recessive? So sex-linked traits, what they are is that they're associated with genes on sex chromosomes. Okay, so before we talked about autosomal inheritance, now we're talking about sex-linked inheritance. So sex-linked traits, these are found in the sex chromosomes, on chromosomes X and Y. And because the X chromosome is larger than the Y chromosome, X-linked traits are more common than Y-linked traits. So therefore, men are more susceptible to sex-linked than women. X-linked dominant inheritance, sometimes referred to an X-linked dominance, is when a mode of genetic inheritance in which a dominant gene is carried out on the X chromosome, whereas X-linked recessive is the same thing, but it's just recessive genes. Okay. And the last one you guys need to know is mitochondrial DNA inheritance. So mitochondria have their own DNA. And the key kind of thing here is that like it's always passed down from your mother. So if your mother has that genetic mutation in their mitochondrial DNA, then they're going to pass it on to all their children. So you can see here, number 2, the mother here has a mitochondrial issue, and therefore she's passed it on to all her children. Okay. And then if you're a female, if you're a mother, then you're going to pass it down to your children as well. Okay. So therefore, these can always be traced through families quite easily. Because if you know a bit about mitochondria always pass down from mother up to the egg cell. Okay. So you always get your mitochondria from your mother. Therefore, your mother has a mitochondrial genetic issue, then you're going to have it as well. Okay. So mitochondrial inheritance is the last one you guys need to know for inheritance. Right. Genetic mutations. This one's even easier. Okay. So genetic mutations, there's only a couple you guys need to know. Silent, missense, nonsense, and frame shift. So silent is when there's no effect in the organism's phenotype. So if you get a change in, say, one nucleotide, say from an adenine to a thymine, and that gives you the same, gives you a codon which codes for the same amino acid, and you don't get a change in your amino acid, so nothing has changed in your polypeptide chain, then that's a silent mutation. A missense mutation is when that change in that nucleotide has caused a change in the amino acid, and therefore you get a new polypeptide chain. Nonsense is when you get a change in an amino acid, and that results in a stop codon, and therefore that cuts your polypeptide chain shorter. Okay, so that's nonsense. And then frame shift is where you either add a nucleotide or a few nucleotides, delete a nucleotide or add a new couple of nucleotides, or duplicate. And that shifts your whole sequence back and forth. And that causes a lot of mutational problems. Okay. So those are the 4 different types of mutations you guys need to know. Silent, missense, nonsense, frame shift. I think silent and frame shift are quite easy to recognize. To tell, if you can't remember which one's missense and which one's nonsense, just think of that like nonsense is like worse than missense. It's like nonsense. It makes no sense at all. Okay. And that's caused by a stop codon. All right. What about aneuploidy? What is an aneuploidy? So aneuploidy is when you get a presence of an abnormal number of chromosomes. There might be like 47, 48, 49, 45, 44, 43, whatever. Those abnormal numbers, that's an aneuploidy. Polyploidy is when you have 3N or 4N or 5N, whatever. It's when you have like 69 chromosomes or like 23 chromosomes. Okay, so normally we have 46. That's polyploidy. What about monosomy, trisomy, and tetrasomy? Monosomy is just when you have one less. Trisomy is when you have one more, and tetrasomy is when you have 2 more. Okay, so monosomy is 45, trisomy is 47, and then tetrasomy is 48. Okay, and the ones specifically the diseases that you the syndromes that you guys need to know about are Patau's, Edwards, and Down. So Patau is trisomy 13, Edwards is trisomy 18, and Down is trisomy 21. I'm not sure I'm allowed to say this, but hopefully I don't get into too much trouble about this. But the way that I remember PSP is teaching us for how to remember 13 is Patau, 18 is Edwards, and 21 is Downs, is that it spells out PED. So like PED, like pedophile, you go from small to large. Okay. 13, 18, 21, PED. Okay. Hopefully I didn't get into trouble for that. If I did, Pavine, please let me know. But that's how I was taught to. Okay. So the PSPs in previous years told us that. So hopefully that helps you guys remember that. All right. What about sex chromosomes or aneuploidy? Klinefelter and Turner's. Those are the 2 you need to know. So Klinefelter is XXY, whereas Turner is X. Okay, so Klinefelter, you've got an extra X, whereas Turner, you've lost a Y. And some of the things you need to know are that in Klinefelter, you've got these sort of syndromes, whereas Turner, you've got these ones here. Okay. Treatment is actually quite good to know. So for Klinefelter treatment, you give testosterone, okay, because you've got a extra X chromosome. And whereas in Turner's, you give growth hormone, estrogen replacement therapy from adolescence. Okay, those are the key kind of things there. In terms of chromosome structure, there's 4: metacentric, submetacentric, acrocentric, and telecentric. Metacentric, if you look at the diagram here, is when the centrosome is in the middle. Submetacentric is when it's slightly off the middle. Acrocentric is even more off the middle, and telecentric is pretty much just on the ends. Okay. So it's talking about the location of that centrosome: metacentric, submetacentric, acrocentric, telecentric. Okay. In terms of structural abnormalities, you got deletions, duplications, translocations, and then in translocations you've got reciprocal or Robertsonian, and you got inversions too. So deletions, you go is when you delete a portion of your chromosome. So if you look at the diagram here, you've deleted a whole section. Duplication is when you duplicate, right? When you double something. You can see here on the diagram. Translocation is when you when you move a section of the chromosome into a different chromosome. So you can see here where they've done swap. And in that you've got reciprocal and Robertsonian. Reciprocal is when you flip it, okay? Whereas Robertsonian is when you join them, okay? Then inversions is when you just invert it, okay? When you've just flipped it around. So those are the structural abnormalities you guys need to know about, and it's all right there. Good. Population genetics. So what are the ones you need to know here? You need to know phenotypic frequency, genotypic frequency, and allele frequency. So phenotypic frequency is the proportion of the population expressing that phenotype. Genotypic frequency is the proportion of the population expressing that genotype. And allele frequency is the proportion of that allele in the population, okay? And I put in the formulas here that you guys need to know in order to calculate those ones. But if you've done methods specialists, I'm sure this is really comfortable for you guys, okay? Should be okay. What about the Hardy-Weinberg law? What is that talking about? So the Hardy-Weinberg law is a theory that they said that it's if there's mating in the population that's random and there's a very large population, then your genotype and allele frequencies will remain constant because they're going to be in equilibrium. So what they're saying is that those genotype and allele frequencies won't change if there's random and large populations mating. But we know that this doesn't happen. We know that mating is not random, and it's very hard for things like this to actually occur because there's things like mutation, migration, non-random mating, inbreeding, assortative mating, genetic drift, bottleneck effect, founder effect. All these things actually tilt that equilibrium, and then that actually stuffs it up. Okay, that's why this Hardy-Weinberg law is only a theory, and it doesn't really work out. But there's a couple of equations you guys need to know. So P is the representative of the frequency of the dominant allele in a population, whereas Q is the frequency of the recessive allele. So the formula, I'm pretty sure it's a quadratic equation, really. Just P squared plus 2BQ plus Q squared is equal to 1. So, and P plus Q is equal to 1. So if you remember these two equations, you're going to be able to work out a lot of the maths that they give you, okay? P squared, 2PQ plus Q squared is equal to 1. P plus Q is equal to 1, okay? And then simple algebra will get you there, right? Good. And lastly, the last thing you guys need to know is biotechnology. So one of the things, recombinant DNA, what is that? So recombinant DNA is, it's formed artificially, and it's by combining DNA from different organisms. So it's generally made from plasmids. And what happens is that we use something called restriction enzymes. They cut out a section of the plasmid, creating these things called sticky ends. And those sticky ends allow inputting of new DNA, okay? And then we use DNA ligase to build up that phosphate backbone again. So we do this because we can then input a certain gene, for example, an insulin gene inside the plasmid, and then the plasmid is able to replicate that gene and create a lot of that insulin. So that's why it's good, right? So you'll have heard of things like when we want to mass produce insulin, what we do is actually we do a lot of this recombinant DNA. We put that insulin gene inside this bacteria, and then that bacteria, because it's able to like replicate heaps in a matter of minutes, is going to keep producing transcription and translation of that insulin gene. And then we get heaps of insulin, and then we're able to take up or like filter out that insulin and then give it to patients, okay? What is PCR, or polymerase chain reaction? PCR is the thing that you use to make lots of copies of DNA. And there's 3 steps you need to know: denaturation, annealing, and polymerization, or extension, okay, whichever one you prefer. You need to know what degrees this happens at. So denaturation is when they separate 2 strands. That occurs at 95 degrees. Annealing is when you reduce the temperature to 55, and that allows primers to join. And polymerization is at 72, okay? And that allows Taq polymerase to basically synthesize that new DNA. Good, and you can see a diagram here and below. You can see DNA primer, nucleotides and whatnot, denaturation, annealing, extension. And that's pretty much all you need to know for that one there, okay? Gel electrophoresis, this is a pretty good one. What it does is that it just separates DNA according to the size of that DNA. So you normally break down DNA in terms of sections and whatnot, and all it does is that it is able to move that DNA through this gel, depending on size. So bigger and longer sort of bands of DNA wouldn't move too far, whereas shorter bands will move really far away. So the way I learned this was that the gel is kind of like a massive kind of like maze or like a jungle. And if you're quite big, it's kind of like a fat man racing a skinny man, right? A fat man's going to be going through that jungle much slower because they can't get through those maze, right? All those tripwires and whatnot. Whereas a small man or skinny man can get through that quite quickly. So smaller bands are going to be further away, whereas longer bands or bigger bands are going to be closer. Wouldn't have moved as much, okay? And the reason behind why the bands are actually moving, the DNA bands are actually moving, is because towards the little where you put the DNA in, that there has a negative charge, whereas the other end has a positive charge. So that actually moves the DNA because DNA is actually negatively charged from its phosphate molecules. So DNA, because it's negatively charged with phosphate molecules, is going to be repelled by that negative charge and attracted by that positive charge. Therefore, it's going to move from where it starts to the other end, okay? And that's pretty much all you need to know for gel electrophoresis. And lastly, what is gene editing? Gene editing is all it's known as is CRISPR. You don't need to know how it actually happens or what actually happens. You just need to know that it's able to cut out sections of genes and then put in new sections of gene. Some of the issues are like cancer risk, stimulating immune response, and it's not very viable long term. And that's pretty much what we need to know for CRISPR, really, or gene editing. Yeah, that's pretty much what we need to know for CRISPR. Good. That was quick and short. Hopefully that was good revision for a lot of you guys. And I'll pass it on to Bhavan.
Thank you, Eric. That was amazing as well. Thanks to our 3 revision lecturers for today. We just got Eric with us, so thanks to Eric right now, but thanks to Yaja and Yash as well for presenting their revision lectures earlier today. It was great to see so many of you guys get around this. As always, the recordings are going to go up on our YouTube page and it'll also be on the Metal page. Metal's also gotten some new additions for you guys, so we put up some assignment breakdowns, well, for the one that you guys have currently, such as ethics and like PSB links and stuff like that. So hopefully you guys are making use of that and hopefully you're able to use them. But otherwise, I hope you guys have a good day. Feel free to reach out with any questions whatsoever. But yeah, thank you guys for coming.
Thanks, everyone.